Well, hello everyone, and welcome to the Global Mosquito Resistance Management Summit. Uh, my team and I have been working for months to get to this event today. My name is Carolina, and I will be your host for today, along with a number of my team that are behind cameras. You just heard Banu and Leanne and Chris, and I will briefly introduce our explain more of how they are going to support our webinar today in a few minutes. First of all, I would like to thank our esteemed panelists for accepting to dedicate hours of their busy schedules to have a technical exchange with us and support this discussion. Our goal is to strengthen our understanding of insecticide resistance, as well as our ability to use control tools against mosquito populations based on science and actual evidence. I can assure you all the content today is just amazing. Our meeting today has an international audience. Hello, everybody, wherever you are. Thank you for joining us. The last time we checked, we had 662 registrants, though we have about 325 participants right now. We hope that the rest of those registrants can join us soon. We also have 315 organizations representing 31 countries. So thank you again for joining.
Okay, so let's start a bit early. What do you say? Should we start?
Yeah, so we got 3 more minutes, I guess, but yeah, we can start. That's okay with you.
Okay, so I will introduce our first speaker of the webinar. His name is Nicholas Delisi. He's an entomologist and laboratory manager of St. Tammany Parish Mosquito Abatement in Louisiana. Nicholas, thank you very much for kicking off our webinar. Please go ahead.
So one important thing, so for all the panelists, you have to stop sharing your screen before the others can share their screen. So, all right.
So I am here. However, I recorded a presentation because I'm not actually able to present by myself where I'm at right now. So if you guys have my PowerPoint pulled up and could play,
I've got it.
The video.
I'm going to start.
You. I'll be available for questions if there's time after, and I'll be in the roundtable though. So if you want to laugh at me throughout my various jokes, or laugh with me, excuse me, I'll be in attendance and hearing you.
Okay, hold on just a second.
See, it turns out it's good that we had those 3 spare minutes.
Hey everyone, my name is Nick Delisi, if you haven't met me before, and I am the lab manager at St. Tammany Parish Mosquito Abatement, just about 30 miles north of New Orleans in Louisiana. And I'm here to talk today about resistance, its mechanisms, and how not mostly to start a statewide surveillance program for resistance. Let's begin with the basics of what insecticide resistance is. I'm sure everyone is very familiar with it at this point, but just in case you aren't, the way that I describe it is a loss of insecticide efficacy due to selection of heritable traits, which is a lot of gibberish, so let's go through an example. So right now in your population, you have resistant mosquitoes. You might not think that you do, but they are there just because of random mutation and chance. So I'm going to characterize those mosquitoes. They are going to be our green ones. You can see they make up the minority compared to these black ones, which are going to be susceptible mosquitoes, ones that will die if they're treated with an insecticide. What happens is they're very rare in our original population, but as we create selection events, as we force them down a particular lineage, in this case by treating with an insecticide, we get rid of most of the susceptible mosquitoes, but you can see compared to the now reduced population size, our resistant mosquitoes make up a larger proportion, which really gets amplified after the next generation of breeding. We see that there's now that ratio of resistant to susceptible mosquitoes has been maintained, and when we go out and treat the next time with the same insecticide, even more of those resistant mosquitoes survive, as opposed to most of the susceptibles now being gone. So they breed one more time, and after multiple treatments, this doesn't happen overnight, but it can happen relatively quickly. We have a population that is primarily composed of resistant individuals. I'd like to start today by talking about how to detect resistance. The main one that I'm going to focus on is the topical bioassay, which is what we do in our parish. There's also the genetic assay, the most famous of which is Kdr, knockdown resistance. Acetylcholinesterase also happens as well, but we won't be going into that today. And finally, the bottle bioassay, which I know everyone is familiar with. You'll hear more about that in other talks, I'm sure, but not so much in this one. We're going to emphasize the topical because that is what I'm most familiar with. So if you're not familiar with the topical bioassay, it is a test where we apply a small dollop of insecticide to the thorax of the mosquito using this very small syringe. And it measures a very particular amount in grams of insecticide to kill your average mosquito, also known as the LD50. This is a little bit different than an LC50, a lethal concentration for the average of the population, because we know precisely how much insecticide this mosquito was exposed to, as opposed to a concentration. This is a brief video that we recorded a couple years ago, so I cringe at some of the things we now do differently, but this is how we do topicals. We'll knock them out briefly, we'll get them on a cold plate, extract some of the insecticide and solvent. This is me making sure that each click of our pipette is consistent. We'll grab a chilled mosquito and expose it to, I think it's something like 0.05 microliters of insecticide in solution. You can see this is a range of cups with different concentrations, or rather, and this is us weighing out the mosquitoes. It's very important to mass standardize everything, as larger mosquitoes are more capable of digesting smaller amounts of insecticide. You can see that they get cups in an incubator. I record some of our results. And this goes by quick, but there's a lot of dead mosquitoes on the left in our higher dosages, as opposed to the controls and lower concentrations on the right. So this is the data that we get at the end, and this is called a dose response line. It's complicated, I won't get into everything, but along the x-axis down here, we have dosage in nanograms of NALED in this case, or whatever your active ingredient is, per milligram of mosquito. This is logarithmic, so you can see it increases very quickly, and then percent dead on the x-axis, or the y-axis. And then this line here represents the LD50, the amount of insecticide it takes to kill your average mosquito from this population, the 50th percentile. In this case, it's 3.1. This does go how we would expect. The lower doses over on the left do have lower mortality than your higher doses over on the right. However, this by itself isn't very useful. It takes 3 grams or 3 nanograms of NALED to kill an average mosquito. Where this becomes really important is when you have multiple populations. So this is that same line that you just saw in blue, but we've added another line for reference in green. This blue line is a susceptible lab strain of Culex squinks known as the Sebring population. This green one is one of our wild colonies that we've caught, I believe, in Slidell, Louisiana. And you can see that there's a pretty big valley between the two. It takes 3 nanograms to kill your average Sebring and about 50 to kill your average wild mosquito. This difference, the proportional difference between these two, is known as the resistance ratio. In this case, it's about a 20-fold difference between our susceptible population and our wild population. We want that number to be as small as it can be. 20 is not exactly great. So in St. Tammany, and in summary for the topical end of this talk, our average quinks require about 20 times more NALED to kill. That number is increasing every year. It's now significantly more than that. This slide was written about a year ago. And as of about a year ago, 200 times more resmethrin to kill. That is a number that is approaching functional resistance as opposed to just a neat, scary finding that you have in the lab. So as a result of that, we have led to a couple of operational changes. We've introduced deltamethrin into our rotation. It still is a pyrethroid, unlike resmethrin, but at least it is a type 2 and has a slightly different mechanism of action, which we're going to talk more about mechanisms shortly. We've increased spray thresholds, so we're spraying a little less frequently, and we're also spraying less frequently because we require more time between our treatment intervals. So let's take a little bit of a shift and talk about resistance mechanisms. This is what underlies resistance in the first place, and they are random mutations that gave rise to resistant phenotypes or expressions of genetic changes. There's 3 different ones: target site mutations, amplified detoxification, and cuticle penetration. Let's begin with target site mutations. These are individual nucleotide mutations, or I think very occasionally locus of mutations, that result in changes in quaternary or tertiary protein structure. So if you remember back to biology class, you have something that's affecting change, your insecticide, and you have the receptor with which it binds to. Well, it binds normally very closely, but the moment you modify the receptor's shape just a little bit, and it can no longer accept that insecticide, the insecticide no longer functions. This is thought to be the primary means of pyrethroid resistance in 80 species, although there's plenty of evidence that there's these others as well. Amplified detoxification is exactly what it sounds like. It is also known as enzymatic resistance, and it is all about the mosquito producing enough enzymes to break down the insecticides it's receiving in order to sequester and excrete it such that it has no effect. And this is largely thought to be the primary mode of resistance in Culex mosquitoes. It certainly is in our mosquitoes, at least in St. Tammany. And finally, cuticle penetration. This one is kind of the most rare, or at least the least observed in nature as far as mosquitoes go. And it has to do with poor absorption due to the cuticle either hardening, getting larger, or just having a slightly different composition, which makes it so the insecticide doesn't penetrate as well. Like I said, this is not particularly common, and I doubt you'll hear a whole lot about this today. Otherwise, I would be very surprised. Let's talk about Kdr, one of those specific base pair mutations. Kdr, if you aren't familiar, is known as knockdown resistance, and that's because when these adulticides are used and you observe the mosquitoes and the effects, it causes temporary knockdown instead of death. So you might look after 5 minutes post-application and see that, hey, everything is dead. But if you come back a few hours later, you'll find that everyone is alive and well, unfortunately. It has a genetic origin at a specific set of base pairs on the sodium ion channel, where pyrethroids and a few other insecticides function. And specifically, those mutations prevent the binding of pyrethroids. Like I say, they change the target site composition just enough that insecticides will no longer bind. The 2 most important, or at least the most studied, are a valine to isoleucine shift at codon 1016, as well as a phenylalanine to cysteine shift at 1534. There are many more of them, particularly if you look outside of 80 species, but those are the primary ones that have been studied in the literature. The one that I want to focus a little bit more on, because it's closer to home, this is what we find a lot more frequently in St. Tammany Parish, at least, is enzyme detoxification. We do detoxification in our bodies, primarily in the liver. I'm not a human anatomist, so I hope that I've got the right organ right there. But basically, what happens, here's a very bad analogy for humans and then mosquitoes. We get a lot of bad stuff. Say you go out drinking one night, you've got a lot of alcohol in your body. Your liver recognizes, hey, this stuff is going to hurt you if it sticks around, so it breaks it down. In a weird world where evolution occurs very rapidly, what could theoretically happen, let's say the air composition changes, there's more toxic stuff in our air that we breathe, something might occur which causes selection for us to develop another liver. I know it's hidden by my picture, but something could happen over the eons that say, hey, in this animal that's exposed to a whole lot of bad stuff, maybe they need a second liver. That's probably not going to happen in humans, but it very well is something that has, I'm sure, occurred throughout history in other organisms. If we change that slightly and make this maybe a little bit more realistic, in mosquitoes and at insects in large, where you have a whole lot of generations rapidly, what can occur is selection for amplified enzyme activity. So maybe they're not growing a second liver because they don't have one, but it very well could occur that their enzyme activity gets ramped up. At the downside of something else, maybe they're worse flyers, maybe they have less energy for something else. But in this instance, if we kind of take it through the liver example from earlier, if there's a bunch of organophosphates that these mosquitoes are being exposed to, selection could occur such that only the mosquitoes that are able to sufficiently break down that organophosphate are the ones that are surviving and reproducing. The way that we test for this in our lab is through a spectrophotometric assay. We call it an esterase assay. It's specifically looking at alpha esterases. We're using alpha-naphthyl acetate as a stand-in for an ester insecticide, which a lot of pyrethroids and OPs consist of. And this is what it looks like. So the deeper the color, so all of these wells in this 96 well plate contain some mosquito homogenate. We ground up mosquitoes and put their guts in here, as well as we're introducing a standard substrate that it could break down and see how well the mosquito does at breaking that down. The deeper the color, you can see these really dark purple colors here, the more esterase activity that we find. So the more insecticide breakdown occurs in these deeper, darker wells. So the result of this might be something along the lines of this. This is what we found from a study back in 2021 in St. Tammany, looking at esterase activity in different regions around our parish. So along the x-axis, we have different populations from across our parish. Sebring is that population that I mentioned earlier that's a lab susceptible colony. They're very wimpy. We would expect that they have low esterase activity. The y-axis, don't worry about the unit because it's horrible, but this is the amount of enzyme detoxification activity, specifically esterases in this case. You can see that there's a few populations, Slemmer and Covington, for example, that have a very wide variance. There's certainly mosquitoes that have high activity, but there's also ones that have low. But then you look at places like Lacombe and Ravine, which I believe is also in Covington, and you see that they are significantly different. There's very little overlap between the Sebring population and the 95% confidence intervals for either of these populations. So we're seeing amplified detoxification in at least a few of our populations in St. Tammany. To wrap up this part of the talk, we're going to kind of change directions after this. Resistance is already out there. You might think, no, we don't have resistant mosquitoes in my district. You do, you just might not have very many of them, although I would argue you probably have more than you think. The real question isn't, is resistance there or not? It's how well are you selecting for it and curbing its development over time. If you have a large Aedes aegypti population or Aedes albopictus, and that's what you do a lot of your spraying for, KDR is a likely suspect as far as what you might need to be looking into as far as mechanisms go. If you find plenty of resistance with KDR, your solution might be to rotate with an organophosphate. We don't tend to see as much KDR effect on OP usage. If you have a lot of Culex mosquitoes like we do and you have Culex resistance, there's a good likelihood that enzymatic detox is what's to blame. And the solution, similar to your Aedes species, is to rotate. Use a different mechanism, or if you have to keep using that same pyrethroid, at least introduce a synergist to minimize the ability for enzymes to break down that insecticide. And finally, mosquitoes don't know any borders. Even if you're doing everything right, that doesn't mean that agriculture down the road is performing everything up to snuff, or that your neighbors might not be having the same problems, which I think is a good lead-in to the LMCA, Louisiana Mosquito Control Association, statewide resistance program. I'd like to just spend a few minutes on this as the committee head for this program in Louisiana. Our intent is to build a statewide resistance database. This is useful for historical reasons to be able to see how things change over time. It also informs if your neighbor's mosquitoes might be hurting your own control efforts, like I mentioned before. And secondly, and probably more importantly, to provide resistance data for smaller parishes that maybe don't have the funding or the skill set to do these kinds of tests. Currently, it is funded by the state. Our Department of Health has granted us grant money for the last couple of years, and we hope going forward. It is performed at a central university to kind of make it as convenient as possible for remote districts to participate. This is currently at LSU, Louisiana State University, in East Baton Rouge. And they historically had done Kdr genetic testing. They currently do bottle bioassays, and all of this testing was only performed on Culex squinks. We might expand that to 80s in the future, but we've got a whole lot of Culex issues in Louisiana. So to touch on our Kdr participation, we had over 9,000 mosquitoes tested in 2020, the first year of Kdr testing. And it was a huge success until in 2021, we had 0 mosquitoes tested. And that was intentional and unfortunate because the USDA out of Gainesville in Florida, who we were closely conspiring with to get all of this set up, published some research saying, hey, Culex resistance and Kdr are poorly linked. When we see high rates of Kdr mutations, we don't necessarily see high phenotype resistance. So as a result of that, we culled it. It's not useful to have a test that's somewhat expensive and laborious if there's a very poor link. As I mentioned earlier, Culex resistance appears to favor enzymatic mechanisms. That's what we find in our parish, and that is backed up by what the USDA found. There are exceptions, of course. We had about 30 bottle bioassays performed in 2020, which was not great for our 60 some odd parishes in Louisiana, and that dropped to less than 10 in 2021. Not a huge success. It's apparent why. It requires a lot more labor. You don't just ship in pools of dead mosquitoes. You actually have to go out and collect living mosquitoes, rear them, deliver them potentially a few hours away to the central location, which has led to a couple of major changes in how we're doing it in the coming years, considering this lack of participation. What we're doing this year into next year is contracting with a local university to hire out some of their students for insect collection, rearing, and delivery. The plan is for them to visit a couple of parishes per week in May through July and retrieve either larvae if they can find them, or at least egg grafts if nothing else, and then bring them back for the bottle bioassay. In the future, I would love to use topical bioassays. I think it's a much more sensitive and discrete test than the bottle bioassay, but I'm more than happy to use any test that we can use to get some resistance data from across the state. And secondly, I'd love to be able to test for enzymes. So this will give the people who are getting this data a little bit more direction, as opposed to just saying, hey, you've got resistance problems, good luck. This says you've got resistance problems, but it looks like this synergist might be a good fit for introduction if you want to get back to normal. So some takeaways: don't start an in-depth new collaboration during a pandemic. I don't recommend it. There were definitely some problems there. Participation is not guaranteed. Shipping worked great for Kdr, but the moment we lost Kdr, it didn't really work well for bottles. And if you want IR data, you're probably going to have to go out and collect it yourself. It's going to cost money. If you're expecting free labor, that is a rarity, is what we've discovered. With that, I'll take any questions at my email, ndelici@stitmad.org, and I look forward to speaking with everyone at the roundtable. Thank you for your time.
Thank you very much, Nick.
You bet.
All right, do we have any time before we move on to the next, or should I save these couple of questions we have for later?
We are right on time.
All right, then I'll save these for later, Nick. Are you going to be in the panel discussion later on?
I'll be there.
Excellent, thank you.
Thank you, Nick. And I have the pleasure to introduce our next speaker, Jennifer Henke. She's the laboratory manager of Coachella Valley Mosquito and Vector Control District in California. Please go ahead, Jennifer.
Well, thank you so much, Carolina, for the introduction. And it's a pleasure to speak to you all today. I am talking about a review of resistance in US mosquitoes. So not just my own district, but taking a look at what we're seeing across the country. So looking at the US and looking at mosquito control here, there's two federal agencies that are in charge of a lot of the work that we're doing. The first being the United States Environmental Protection Agency. You'll hear me refer to them as the US EPA. They are the ones that regulate the pesticides and the repellents that are used by both professionals like mosquito control professionals that you're seeing speak today, but also private homeowners that are using repellents to protect themselves from bites. They work very closely with the Centers for Disease Control. We have a speaker later in today's program speaking from there. And the Centers for Disease Control, or the CDC, provides information on insect-borne diseases. And when there's outbreaks, they work very closely with both local and state agencies in order to disrupt those transmission cycles. These two agencies do work closely together, and then they do work closely with local and state groups. But you'll see that the information from one federal agency often references the information that's on a different federal agency. And so the work goes back and forth between the two. So if we look at the US EPA's website, knowing that they're often providing information for members of the public to know why are these mosquito control professionals doing things? What's going on? The US EPA references the diseases that the CDC lists, and the EPA says that the primary diseases in the US are Zika, West Nile virus, malaria, encephalitis, including Eastern equine, Japanese, and St. Louis, yellow fever, dengue fever, and chikungunya fever. And so these are the reasons that mosquito control professionals are making applications. When we look at these diseases and the viruses and pathogens, we can see that we're really looking at two genera of mosquitoes that most of the work is being done on. On the left is Aedes mosquitoes, Aedes aegypti and Aedes albopictus, the vectors that are transmitting the viruses that cause Zika, yellow fever, dengue, and chikungunya. On the right, Culex mosquitoes. Pictured here is Culex pipiens, Culex quinquefasciatus. Over on the far right is Culex tarsalis. These are our vectors of West Nile virus, Eastern equine, Japanese, and St. Louis encephalitis. And so the focus of this presentation is going to cover those two species, those two genera, ignoring the fact that there is work that is done for malaria mosquitoes, for the Anopheles, and for Eastern equine encephalitis, where Culecida plays a primary role in maintaining that transmission of that virus. So where are these mosquitoes in the US? Where can we find them? Well, the CDC has put out a map of probable ranges for Aedes aegypti and Aedes albopictus. This is from 2017. We can see that we're going to primarily find these mosquitoes here in the southeastern part of the US and over in parts of the western states. And again, albopictus is a little bit more cold tolerant, so it's going to be found more northerly than our Aedes aegypti mosquitoes. When we look for probable ranges of the Culex mosquitoes, I've stolen these images from a really fantastic paper that came out last year by Goris of the Suitability of Habitats for Culex mosquitoes in the Americas. So this is Culex tarsalis. We can see that it's found in a big part of the central parts of the US here with these darker colors. All the way almost to the eastern US, but mostly in the Midwest, and then really out here in California where I am and other parts of the western US. And then for the Culex pipiens mosquitoes, I'm lumping pipiens and quinquefasciatus together as part of this discussion. So for pipiens, we can see that we find them in the northwestern US, up here in Washington, and then up in the upper Midwestern states out to the eastern US. And more southerly, we find the Culex quinquefasciatus. So going along the bottom parts of the US, we find large pockets of Culex quinquefasciatus mosquitoes. So in doing a review of what kind of pesticide resistance we may have in an area, a really fantastic resource is the Arthropod pesticide resistance database. If you're looking for any kind of resistance to insects and the pesticides that are being used to control them, it is done in partnership with the Insecticide Resistance Action Committee. Registered users are able to make entries on cases of insecticide resistance for any particular insect, but anyone is able to search the database. And so when you go into search the database, what you can find is that you can put in the order of insects that you're interested in and the family. You can even go down to the genus and the active ingredient, the country, and look for where others have already done some of that work for you to find where there are resistance cases for a particular insect. It is a global database, And so you're able to get entries from anywhere in the world for any particular insect that you're interested in. And looking at the insects that I was interested in for this review, we can find that globally, there are quite a few records of insecticide resistance for Aedes aegypti, nearly 600, and more than 250 for Aedes albopictus. I said I was grouping those Culex pipiens mosquitoes together. You can see that Culex quinquefasciatus had a large number, but there's other records for Culex pipiens, and then fewer for Culex tarsalis. There are records for Culex tritenorincus, not in the US. It is an important vector of the Japanese encephalitis virus. And so I wanted to highlight that for our colleagues that are following that outbreak. So looking at those records, these maps are going to be fairly similar across the next series of slides. I wanted to first highlight that there are lots of entries from work that was done for organochlorines, particularly DDT. And so in this case, what you're going to see is that a mosquito in a particular area means that there was a resistant population that was detected from that territory or that state if there's a box around that particular mosquito. So in this case, this brown box is for organochlorine resistance that was detected in Puerto Rico, Florida, and Texas for Aedes aegypti. When we then look at Culex tarsalis, we see that there was resistance detected in Utah, California, Oregon, and Washington, so our western states. And then for Culex quinquefasciatus pipiens complex, detections for the upper Midwest, upper Northeast, which is New York, Massachusetts, and New Jersey. Detections for both Puerto Rico and Georgia. And then back over to the West, we have Utah, California, and Hawaii. I show you DDT for a couple of reasons. Sometimes US mosquito control is said that we don't do resistance work, but this is a much older work that was done by George Giorgio in many cases. And secondly, we know that organochlorine resistance can be also cross-resistant to pyrethroids. And so we might see that this is an important thing as we're continuing to consider resistance in our mosquitoes today. Currently, by the US EPA, there are two classes of chemicals that are registered for control of adult mosquitoes by mosquito control professionals. On the left is the organophosphates, and on the right is the pyrethroids. For organophosphates, we're talking about chlorpyrifos, malathion, and temifos being some of the big players. For the right, I've included pyrethrins as the natural pyrethrins, which are not the synthetic pyrethroids. But we can see we have both type 1 and type 2 pyrethroids as part of this. There are limitations in the rest of the review that I'm about to show you. So not just using the Arthropod management database, I did search the published scientific literature for other instances and cases of insecticide resistance that were reported. This is very broad at a state level. And we know that resistance, as Nick so eloquently put out, is happening at a population basis, a much finer scale than what I'm showing you today. It does rely on the published information, and many of my colleagues on this call have been doing this kind of work. Unfortunately, sometimes it's not always published. As Nick also pointed out, sometimes we need funding and time to be able to do that kind of work. And so being able to have everything that is going on was a harder task, but I have relied on the published literature. And I also want to highlight that I'm relying on bottle bioassays and field data for this, rather than some of the genetic tests that Nick was pointing out before. I chose that because I wanted to see that there was the mechanism was actually being expressed, not just the genetic capabilities of that mechanism were there. So with those limitations in mind, I'm going to start with Aedes aegypti. We'll go through organophosphates and then pyrethroids next. So for organophosphates, there have been recorded resistant populations of mosquitoes in both Texas and in Puerto Rico, primarily to malathion, but also to temifos. And for pyrethroids, we have seen detections of resistance to a whole suite of different pyrethroids in all of the instances that were found, which were Puerto Rico, Florida, Texas, and as well as New Mexico. So looking at those together and bringing forward that organochlorine, that DDT resistance that's in the more historic literature what we can see is that there's a large concern for the mosquitoes that are Aedes aegypti in Puerto Rico and in Texas, where there's quite a bit of resistance, or at least a resistant population to all three of the different classes of pesticides that we've talked about, as well as there is concern in Florida for that resistance being developed and being passed on year. Looking then at Culex tarsalis in our Western US, we see that there were resistant populations to those organophosphates. Malathion is the primary one here, but also chlorpyrifos for Utah, California, Oregon, and Washington. And then some additional work for pyrethroids for Culex tarsalis in California, where there were resistant populations. You'll note that here in the upper Midwest and South Dakota, there is now a Culex tarsalis that's pictured here. There were susceptibility studies that were done there, and at least in that case, the Culex tarsalis were susceptible to permethrin. So bringing forward our malathion work combined with that older DDT work, we can see that there's concern in California that there's resistance to all three of those classes of pesticides. And again, resistance to two classes in Utah, Oregon, and Washington, knowing that our organic chlorine resistance, that brown box, may mean that there is the opportunity for there to be resistance to those pyrethroids. Finally, moving to our more complex group, because there were a lot more work, as we are aware, Culex pipiens and Culex quinquefasciatus is the vector of West Nile virus, which has been found routinely in the US for the past 20 years. There is a lot more work that's been done on this mosquito, and that's increased quite a bit recently. But there is cause for concerns. There's work that shows that there's resistant populations in the Midwestern, all the way over to the Eastern US. So we've got Minnesota, Illinois, and Pennsylvania having resistant mosquitoes to malathion. We have in the Southeastern US from Georgia all the way over to Texas, resistant populations, primarily malathion, but also Temophos. And then in the Western US, we have resistance in Utah and California, primarily to malathion, but also chlorpyrifos. Moving forward to the pyrethroids, we see a similar pattern when it comes to our mid region of the country of Minnesota and Illinois and Pennsylvania, also seeing a suite of resistance to etofenprox, to permethrin, to pyrethrins. We see that we have resistance moved further up the coast in the southeastern and the central part of the southeastern, so upper to Tennessee and North Carolina and South Carolina, and including down where we were before, skipping over Georgia. We did find susceptible populations in the literature there, but also Utah and California having resistant populations to pyrethroids for Culex pipiens and Culex quinquefasciatus. So bringing forward our DDT results from earlier, most concerning here is these Western states where California and Utah are showing resistance populations to all three of those classes of chemicals, but also concerning are Minnesota, Illinois, and Pennsylvania, where both classes that can be used in the US are showing that there's resistance, as well as in the southeastern part of the US where we're seeing quite a bit of resistance. That's a lot of different information from a lot of different people. And so I went back to try to reclassify that in a way that maybe it's a simpler take home for people about why this is so concerning. So looking at this again, what I've done is that you can see that the left facing mosquito, like this brown one that's here in South Dakota, is our Culex mosquitoes. And the blacker mosquito facing to the right is the Aedes mosquitoes. If there was a report of resistance to DDT, that organochlorine, I've shaded that box around it yellow. If there was resistance reported to one of the two current classes of pesticides that we can use for adult mosquito control, either the organophosphate or the pyrethroid, it's this oranger box. And if there was resistance reported to both of the classes that are currently available, organophosphate and pyrethroid, then it's this redder box. You'll see a few other notes that I'll highlight for you here. There has been some work that's been done on Aedes albopictus. So in a couple of cases, I'm highlighting that there's an albopictus population instead of Aedes aegypti. And CT over on these boxes is indicating that that's Culex dorsalis instead of the Culex pipiens quincefasciatus complex. But the take home that you might expect from this is that there's a fair bit of orange and red on this map, things that are concerning for US mosquito control professionals. Particularly concerning is here in the southeastern US, where there is a lot of mosquito control activity that is done on a routine basis to protect those residents and visitors, but there are populations where there are resistance to one or both products that can be used in that particular area. Also concerning is in the Western US, where we're seeing strong levels of resistance. And we're also concerned, of course, by what's going on in the territory of Puerto Rico and in the state of Hawaii, both areas that receive a lot of visitors on an annual basis. It feels a little bit doom and gloom to end a presentation like this, but I'll try to provide a little bit of sunshine to go with this. Resistance is clearly an issue in US mosquito control. And I know that my colleagues in the audience are probably saying, but it's not driven by us. I get that, but it's still something that we have to deal with. It's the consequence that's out there that is ours to have to work on and to try to make better. More information is needed. You see there's big holes in those maps, and I'm enlightened and encouraged by the work that the CDC has put into the centers of excellence that we have at partnerships of universities working with mosquito control professionals. And as Nick pointed out, funding is needed in order to continue to make this work successful. Resistance is also a finer population level problem than what is shown. So even if you're in one of those states where you saw red, doesn't necessarily mean that your mosquito control products are not going to work. You do have to, as a local level, examine what's going on for those populations and to routinely take a look at that to see what's changing over time. I will highlight that I have a few selected references. If you'd like the full bibliography, my email will be on the next slide. I want to particularly highlight that those beautiful maps of the Culex mosquitoes came from the Goris publication. It's a really lovely piece that includes not just the US, but the rest of the Americas as well. I also want to highlight the work that Stephanie Richards at Eastern Carolina University did with her colleagues. It's a really fantastic study, and it added a lot of different pieces of information that hadn't been part of that Arthropod pesticide resistance database. With that, this is my email. I do have a picture of my colleagues from Indio, California. If you're in the Palm Springs, California area, please drive another 25 miles to the east. We'd love to say hi to you. Thank you so much.
Thank you, Jennifer, for such a comprehensive review. We do have 2 minutes if anybody wants to share a question in 2 minutes. Please raise your hand.
There were, I do
Yes.
have one, I do have one question that popped up in the Q&A for Jennifer we can quickly touch. Says, thank you, Jennifer, for the review. Please, is monitoring in the US done all year round or at what intervals?
Sure, it's a great question. What I was showing is work that's published, and so some agencies are doing it on an annual basis where they go through and check their population once a year. In some cases, it's the only report because it was a graduate student that was doing some work and maybe collected mosquitoes for a couple of years and then didn't do any additional work because there just isn't the funding for that. So it's really varied and it's really inconsistent, and it would be great to have additional funding to make that a more consistent annual basis that everybody can do.
Thank you, Jennifer. Okay, I'm going to go ahead and introduce our next speaker. We have the pleasure of having Dr. Charles Wonji. He's a professor of vector biology and genetics at the Liverpool School of Tropical Medicine, and he's also executive director of the Center for Research in Infectious Diseases in Cameroon. Please welcome Dr. Charles Wonji. Go ahead.
I'm Charles Wonji, and I'll be talking about the molecular drivers of insecticide resistance in malaria vectors. It's important to start by mentioning that vector control really plays a major role in the prevention of most vector-borne diseases. And this is done usually through long-lasting insecticide nets, LINs, and indoor residual spraying where insecticide are sprayed on the walls or live residing. And at the bottom here, I just want to ensure that vector control have really played a major role in reducing the burden of malaria since the year 2000. And as you can see on your right, the contributed by 70% to the reduction in malaria cases worldwide. But we are facing a major challenge because there is a growing and widespread report of insecticide resistance in most malaria vectors. As you can see, this is the situation before 2004, where if you look at the African continent, more dots were green, meaning that vectors were susceptible. But back in 2015, what we have is more red dots showing really widespread resistance. And on your right, I put a picture there that I took myself in Uganda, where mosquitoes, fully blood-fed, were actually resting inside the bed net supposed to kill them. Why? Because of resistance. What we need to know is that, unfortunately, there are only limited insecticide classes that could be used for the control of public health pests. We have 4. Among them, organochlorines with DDT, organophosphates. One example is temephos. We have carbamate. One example here is propoxol, but we also have bendaocap. We have pyrethroid with type 1 pyrethroid. One example is permethrin. Type 2 example, deltamethrin. And it's good to mention here that pyrethroids are the most used for control program. And the reason is they have the lowest toxicity to humans. And that's why most bed nets actually contains pyrethroid. All these insecticides, what they do is to target the insect nervous system. And if you take the case of DDT and pyrethroid, they both bind to the sodium channel. And as you can see on your right, the model displayed there, what they do is that they keep the channel in an open state. And what happens is that they cause repetitive firing of neurons, leading to paralysis and death of the insects. For organophosphates and carbamates, they rather bind to the acetylcholinesterase, and they probably produce the same effect, also leading to death of the insects. I just put there a model of acetylcholinesterase docked with an insecticide. So what is the genetic basis of resistance? What we need to know is that often the wild population is susceptible before the insecticide is applied. But as we apply the insecticides, the few resistant that were in that population will be selected, and the proportion will increase naturally. And the resistant insect, therefore, will be at an advantage, and they will reproduce, and the genetic changes that confer resistance will be transferred from parents to offspring, and resistant insects will become more and more predominant. As you can see in the cartoon here, as we apply insecticides, the proportion of resistant allele increases significantly. And this will even go quickly if the allele is dominant, and slower if the allele is recessive. And if the resistance is actually dominant, as it has been seen in the population, in a very short number of generations, we can move from susceptible to resistant population. Another question is, what are the sources selecting resistance in the field? One of these is agricultural practices. Why? Because the same insecticides used in agricultural sector is also used in public health. And that makes that insecticide used by farmers actually contaminate vectors, breeding sites, and that will induce resistance even before these insecticides are even used in the public health sector. And that's why there needs to be a strong collaboration between agricultural sector and the public health sector to improve the management of those existing and few insecticide classes. Now, why should we bother about resistance in public health? Does it really matter? I think I will say yes, by just showing this slide where through a modeling, WHO predicted, for example, that if we are not careful managing resistance, that may lead to a significant increased mortality of up to 260,000 deaths a year if nothing is done. So it's not just an academic issue, it's really something that can impact public health seriously. Now, let's talk about mechanism of resistance. We have overall 4 main mechanisms. We have what we call the target site, where the target of the insecticide is modified, preventing insects to bind, and therefore it will not produce the lethal effect expected. We also have behavioral resistance, when mosquitoes change the behavior to avoid exposure to insecticides. We have what we call reduced penetration. As you can see there, the insect will thicken the cuticle, limiting the amount of insecticide that may go in, and therefore mosquito will be able to survive. We also, and one of the major mechanisms is what we call metabolic resistance. And in this case, resistant insects are able to produce large amount of enzymes that are able to break down the insecticides before it reaches the target site. So these are the 4 main mechanisms of resistance. Let's start with the biochemical resistance mechanisms. We focus on these 2, target site, metabolic resistance. For target sites, we have various types. This one is what we call the knockdown resistance mechanism. And this type is actually caused by a single amino acid change that occur in the gene called sodium channel transmembrane protein. And that leads to the Kdr type of resistance, knockdown resistance. These mutations are actually able to confer resistance to pyrethroid and Ddt resistance. Leucine to phenylalanine substitutions is really something widespread concerning this mutation. But we have to bear in mind that not all species have this type of mechanism. So it's not always expected that all mosquitoes will express the mechanism. For this, there are simple DNA-based diagnostics tools allowing us to actually easily detect the resistance in the field. The other target site is called altered acetylcholinesterase. And in this case, we have a glycine which is replaced by a serine at position 119 of the gene, and confer resistance to organophosphates and carbamate. And again, not all species of mosquitoes will have it, because other species like Anopheles funestus, we have a different amino acid change, asparagine to isoleucine at codon 485. So we always need to bear in mind that things are not standard. Now let's look at the metabolic resistance, which is one of the major ones, as I mentioned, because it's more likely to lead to operational control failure. That's why we pay attention to this mechanism significantly. And it's also a bit more complex because it's not contrary to target site. You don't have one enzyme involved. You have, for example, 3 major enzyme families involved besides small ones. And the major ones are cytochrome P450 monooxygenases, glutathione S transferases, esterases. And the reason why this mechanism is complex because there are many enzymes potentially involved. We are talking about maybe 200 enzymes. And the challenge is to know which one is really driving resistance. And even if you find the one that is associated with resistance, the other challenge is that there are other mechanisms through which that single gene may confer resistance that may occur by a point mutation in the coding region. It might be mutation in the regulatory loci or the transcription factors. So there are many mechanisms making this mechanism very complex. But what I would like to share with you now is that we have made significant progress in detecting this resistance mechanism, starting by detecting the major genes which are overexpressed when we talk about pyrethroid resistance. For example, the several genes, P450 genes like sub6P3, sub6M2 have been detected as playing a major role in Anopheles gambiae. This has been done using transcriptomic, using functional genomics to detect those genes. And also concerning other species like Anopheles spinosus, another major malaria vectors in Africa, using transcriptomics based on RNA-seq methods. Major genes have been detected across the continent. But something interesting was that, you know, mosquitoes from different regions of Africa, they don't exhibit the same genes to be resistant. So making it even more complex, because what's happened in one, let's say in Central Africa, is not the same in Southern or East Africa. So that means that it's not one size fits all in terms of implementing control program. What about detecting the specific genetic variants which are conferring a metabolic resistance? Progress also has been made there. And in the slide we have here, I'm presenting the case of the first DNA-based metabolic resistance that we detected. And this was in a gene called glutathione S-transferase epsilon 2, where we detected a single amino acid change from leucine to phenylalanine at position 119, which was actually acting by enlarging the substrate binding pocket, allowing more insecticides to get in and to be metabolized. We show that this amino acid change was strongly linked with the phenotype, those having it able to survive more than those without it. Using that, we designed a simple DNA-based assay, allowing us to crack the spray of resistance, which was located mainly in West Central and East Africa, not in the Southern Africa. And using this expertise, we move forward to tackle the case of cytochrome P450, detecting also a specific polymorphisms which are tightly linked to the resistance phenotype. And we use that information to design simple DNA-based assay, allowing also to crack resistance. What we did was to first of all confirm that the polymorphism was really correlating with resistance. And indeed, we show that once mosquito have that polymorphisms, it's able to survive exposure to pyrethroids. That was done for the first gene, but also for another P450 genes, confirming that yes, DNA-based markers for P450-based resistance could be detected in malaria vectors. And now that we had such tools, we could answer key operational questions like, what is actually the impact that metabolic resistance is having on the efficacy of bed nets? And in Cameroon, we built experimental huts, mimicking a situation where people are sleeping under the bed nets. We can release mosquitoes there with different genotypes and compare those that have survived exposure to bed nets, those that are blood fed, to find out what was the impact of the genotype. Overall, what we show through that experiment is that when mosquitoes have the homozygote resistance allele, are homozygote for the resistant allele, they are able to survive better when exposed to pyrethroid-only nets. They can blood feed more, meaning that it's reducing the efficacy of bed nets. But the good news was another type of bed nets that we call PBO net, synergist net, because it incorporates a synergist which is able to block the activity of P450. Those nets are actually more efficient even when mosquitoes have the P450 resistant allele, which is showing that in the field, control program should rather use those PBO nets. And because we have another markers, we could answer the question, what's happened when mosquitoes have actually not just one resistant allele, but two? what we show is that there is an additive effect. So if a mosquito is double homozygote resistant, then it will survive much better. Just look here, the double homozygote resistant, they are all alive, whereas the double susceptible tend to be the one dying the most, showing that yes, we need to act before mosquitoes are becoming very resistant. And talking about molecular drivers, there are other factors. Mosquitoes can also become resistant by having what we call increased copy number of certain resistant genes. This has been shown in Anopheles gambiae, where some pyrethroid genes, pyrethroid resistant genes are present in many copies, allowing mosquitoes to survive. And resistance also could come, I mentioned earlier, through cuticular resistance. Mosquito can thicken the cuticle to limit the amount of resistance of insecticides getting in. And progress has been made recently by detecting the genes which are associated, including 1p450 that can help produce cuticular hydrocarbons. But for these mechanisms, we still don't have a molecular marker yet. And to make things even more complex, in end of 2020, it was shown that another mechanisms confer resistance exist, and this is through sensory appendage proteins called SAP, which can confer resistance in other malaria vectors. So we always have to be open-minded in terms of what is the mechanism potentially involved in the field. Overall, when we know what are the specific genetic variants driving resistance, there are key operational questions that we can help answer. One of them is resistance really increasing malaria transmission? We did that by using the GST E2 allele. We actually genotype those mosquitoes infected versus non-infected. As a result, we show that the mosquito infected were mainly homozygote resistant, showing that yes, resistance actually may lead in some cases to increased malaria transmission. And that was further compounded by the fact that those resistant allele, those with the resistant allele were able to live longer. So meaning that that could really increase malaria transmission. Another question was, what is the fitness cost? Because if you want to implement resistance management like rotation, we need to know whether there could be a restoration of resistance, of susceptibility. And indeed, for P450 allele, we show that there is a restoration of resistance. If you don't apply insecticides, the strain may become susceptible over time, which is a good news that if we stop using pyrethroids and switch to another one, in a few years' time, the population may become susceptible again, which is a good news. And we also can look back to see really that using scale-up of intervention actually is driving the selection of resistance. Look, for example, this is in Mozambique. Before bed nets were scaled up, most mosquitoes were susceptible SS, but 15 years later, they are all resistant, showing that bed net is actually driving resistance in the field. But the good news is we are having new tools based on new insecticides like Fludora Fusion using neonicotinoids or new bed nets using chlorophenol. So we need to take advantage of the knowledge we have gathered to better manage these tools and to implement appropriate resistance management to avoid having to deal with population which are now completely resistant. We need to act when it's still resistance is still at the low frequency. And how could we do that? Using methods like mixture, rotation, and mosaic to actually limit the frequency of resistance. To summarize, yes, resistance is a challenge for vector control of malaria, and the metabolic resistance is playing a major role. But the good news is recently we detected new diagnostic tools, and this is now allowing us to do more in the field. And this is actually something that should help us to detect resistance and to manage it properly. So that is really where I will stop here, and thank you for listening.
Thank you, Dr. Wanji. It's a complex subject indeed. Thank you for your presentation. I'm going to introduce our next speaker. So we are fortunate to have Dr. Gabriela Gonzalez Olvera. She works at a collaborative unit for entomological bioassays at the University of Yucatan, Mexico. Some of you received the original agenda had Dr. Adriana Flores, who unfortunately couldn't be here with us today, but we're fortunate to have Dr. Gonzalez. So please welcome Dr. Go ahead, Dr. Gonzalez.
Good morning to all. I am Gabriela Gonzalez Olvera, and first I would like to thank the organizers for inviting me to this talk. I am going to be presenting the surveillance of insecticide resistance and operational research with Aedes aegypti in Mexico, with emphasis in the experience from Yucatan State. Before I begin, I would like to acknowledge the contribution of the following colleagues to the work I will be presenting. This is a large collaborative effort that involves the Federal Ministry of Health of Mexico, the State of Yucatan Ministry of Health, the CDC, and the Universities of Yucatan and Emory. I will start this talk by pointing to the epidemiological and entomological reality of Mexico. With a total of 32 states, Mexico is one of the top countries in the Americas with respect to the number of human dengue cases it reports. The figure on the left shows the dengue case counts during 2000 to 2012 and shows that most of the country, with the exception of the center and north municipalities, have reported cases. This is in big part explained by the widespread distribution of Aedes aegypti, as it is shown in the map of the right. As a consequence of the high human health and economic toll of dengue and other Aedes-borne viruses such as chikungunya and Zika, the Federal Ministry of Health, under its Center for Disease Prevention, CENAPRESE, established a state-of-the-art surveillance platform for the capture and management of entomological and epidemiological information. This online decision support system has 4 main modules, which include surveillance of Aedes aegypti using a network of OV traps, the regular monitoring of insecticide resistance in Aedes aegypti, the detection of dengue, chikungunya, and Zika from field-collected adult mosquitoes, and the surveillance of other vector species such as kissing bugs and ticks. The overall goals of the platform are to use the entomological information to monitor the impact of vector control actions, provide information to guide where control should be tailored, therefore increasing the efficiency of the program and also rapidly detecting the occurrence of insecticide resistance, and use such information to guide the purchase of insecticides to maximize the effectiveness of interventions. I will describe 2 of the components: the OV trap surveillance and insecticide resistant monitoring modules of the system. Mexico has perhaps one of the most comprehensive entomological surveillance programs for Aedes aegypti. A network of 230,000 OV traps located in 32 states and 712 cities are monitored weekly for the presence and number of Aedes aegypti eggs. Such data are routinely used to quantify the density of vectors and to identify areas of high entomological risk for arbovirus transmission. When looked over time, the OV-trap data also allows the Ministry of Health to evaluate the impact of interventions deployed locally. For example, ultra-low volume applications from vehicles within a city, And finally, the ovitraps provide eggs for evaluating susceptibility of the local population to insecticides. This image shows the entomological surveillance platform in action. The dots show the individual ovitraps, which are ranked by color according to the density of Aedes aegypti eggs. Red indicates the highest quartile in the number of eggs for a city. The buffers show a 100-meter area around each positive trap on a given week. Finally, the small red circles show a 100-meter buffer around the location of human cases detected on the same week as the ovitrap monitoring data. Therefore, the Ministry of Health staff can use these risk maps to guide where intervention should be performed. A recent innovation in the entomological surveillance of Mexico has been the establishment of BioSciences units. There are currently 22 units have been staffed by hearing biologists and entomologists who are in charge of rearing Aedes aegypti from ovitraps to have enough female mosquitoes to conduct bioassays. Conduct the standardized CDC bottle bioassays using a list of molecules of all insecticides approved for public health use in Mexico, and conducting these activities every 2 years to inform insecticide procurement. I am responsible for one of these bioassays units, the one in Yucatan State, where the arrow points. Another strength of the program is that all guidelines and procedures are openly available to the states and the public in general. On the left, you can see the latest manual for insecticide resistant monitoring that the Federal Ministry of Health generated. In the year 2016, BioSciences units conducted the first national assessment of insecticide susceptibility in Aedes aegypti, which was published in 2017. I will be reporting findings from this paper, which constitute the latest and most detailed characterization of Aedes aegypti susceptibility in Mexico. These maps show all the localities evaluated as dots, and with colors the results from the CDC bottle bioassays ranked by the WHO convention used to characterize resistance to insecticides. In the map, the red dots indicate mortalities below 90%, which suggest resistance to the active ingredient tested. In yellow are localities where mortality was above 90% and below 88%, considered as indicative of potential resistance. Finally, localities in green indicate susceptibility to the active ingredient. As you can see from the two figures, in the year 2016, Aedes aegypti was fully resistant to type 1 pyrethroids, in this case, permethrin and bifenthrin. Type 2 pyrethroids did not show the high level of resistance as type 1, but the agreement is that resistance is widespread with only a few foci of susceptibility. Given the high level of pyrethroid resistance, Mexico used this information to phase out the use of pyrethroid insecticides for the control of Aedes aegypti. Other insecticide groups, such as organophosphates, also showed widespread resistance, but not as high as for type 2 pyrethroids. Finally, given their limited use, carbamates are still a valid option given the almost absence of resistance in Aedes aegypti. The latest assessment of mechanisms of insecticide resistance at the national level was conducted for pyrethroids by Adriana Flores Group, which characterized the rapid increase in Kdr mutations in Mexico from 1996 to 2008. As seen in the map, the red bars increased dramatically compared to previous years, indicating a rapid increase of the Kdr mutations in the position 1016. In the city of Merida, the increase was very pronounced, and led the Ministry of Health to phase out the use of pyrethroids to control Aedes aegypti starting in the year 2016. This large-scale pattern of rapid and widespread evolution brings up important questions about the reliability of pyrethroids for mosquito control. Specifically, with colleagues from the Collaborative Unit of Entomological Bioassays from the University of Yucatan, Dr. Pablo Manrique Saide, and from Emory University, Dr. Gonzalo Vázquez Procopec, and from the Federal Ministry of Health, Dr. Fabián Correa Morales, we were interested in two specific questions regarding insecticide resistance in the state of Yucatan. The first one was, at a finer scale, which means within the state of Yucatan, how is the distribution pattern of pyrethroid resistance and Kdr mutations in Aedes aegypti? And the second was, given pyrethroid insecticide applications were phased out since 2016 in Yucatan, what drives the maintenance of high levels of resistance and Kdr mutations? We conducted a study in 2016 to address the first question around the city of Mérida in Yucatan. The premise of the study was to quantify the fine-scale variability in phenotypic and Kdr resistance to pyrethroid within nearby towns. The hypothesis being tested was that if pyrethroid resistance and Kdr frequency followed the national trends shown earlier, all endemic towns within the region should have similar trends in resistance. When phenotypic resistance was quantified, marked variability was found, with no specific trend in which town would have high susceptibility given its prior history of dengue transmission. As the figure shows, for instance, Progreso and Concal were closer to each other yet showed marked variation in the percent mortality of Aedes aegypti exposed in CDC bottled bioassays. Even further, both Kdr mutations, the alleles in position 1534 and 1016, showed significant and marked variability among cities, with tests of genetic differentiations identifying lack of homogeneity in their distribution. To investigate the geographic pattern of variability in Kdr even further, we selected one of the towns from the previous study called Unucma, which showed phenotypic susceptibility to delta methrin, and investigate the microscale pattern in Kdr haplotypes within randomly selected city blocks. The hypothesis of the study was that, given there was not a strong signal in resistance when the entire town was characterized, Kdr haplotypes should be similarly low at the microscale. Aedes aegypti adults were collected indoors with Procopac aspirators and individually tested for Kdr mutations in the 1534 and 1016 positions. Haplotypes combining both mutations were generated. This map shows the double resistant haplotype for both Kdr mutations. The study was conducted on the dry and wet season of 2014 to 2016 and covered 4 collection periods. As it can be seen in the map, the pattern of Kdr distribution was contrary to what was hypothesized. In 2014, rates of the double mutant 1534 and 1016 haplotype were highly variable within the town, ranging from 0 to 80% within less than 500 meters. Even more interesting, as the wet season ended, the upcoming dry season showed a rapid increase in the haplotype frequency throughout the city, even in the absence of pyrethroid used by the Ministry of Health. The wet season of 2016 showed reduced frequencies compared to the previous dry season. The dry season of 2016 showed almost invariant rates of the double resistant haplotype. This study allowed us to conclude that even in the absence of pyrethroids for mosquito control, rates of Kdr change as a function of the seasonality of mosquitoes. Other factors may be operating and may help explain why pyrethroids resistance and Kdr haplotype frequency vary both in space and time. This led us to investigate what we hypothesized was a probably driver of Kdr variability in the absence of their use by the Ministry of Health. Household aerosolized space sprays used to control insects. A study conducted in Yucatan characterized household expenditures in commercial aerosolized insecticides and found that the most common insecticide inside houses are aerosol spray cans, such as Raid, which contain a mixture of pyrethroids like D-alethrin and phenolphthalein. People reported using such insecticides primarily for flying insects at a daily or nearly daily frequency. The benefit they see in using these products offsets their cost, which ranged at about $30 per year. Given this high reported use, we hypothesized that it could be that the high frequency and heterogeneity in Kdr within cities may be reflective of variability among households in commercial insecticide use, and that such insecticides may be an additional and a strong pyrethroid selection pressure on Aedes aegypti. To investigate that, we conducted studies in 2018 in experimental houses located in Mérida. These are regular uninhabited houses that UCBE rents long-term for conducting insecticide evaluations and population ecology studies. Specifically for this study, we used individual bedrooms as the unit of experimentation. So we placed mesh enclosures with mosquito to make sure that they would get exposed to the insecticides, and then applied the aerosolized space sprays as directed in the level. And this involved applying in the room the insecticide at a 45-degree angle for 10 seconds, then closing the door and counting mosquitoes, knockdown, and mortality after 20 minutes exposure. The study was repeated using 3 strains with different levels of resistance, collected from the towns we evaluated in the previous study. All dead and alive mosquitoes were individually tested for the Kdr mutations 1016, since previous studies found that the mutation 1534 was presented in 98% of the population. As it can be seen in this plot, when a susceptible Aedes aegypti strain was used, the insecticide killed 100% of mosquitoes. However, when a pyrethroid resistant strain from one of the towns evaluated before was used, mortality was only 30%. This strongly indicates a significant decline in efficacy of the product due to the presence of resistant mosquitoes. Now, we wanted to know if those mosquitoes surviving were resistant or perhaps just did not get impacted by the insecticide. Our findings clearly show that the mortality in the resistant Aedes aegypti strain was a function of their Kdr frequency. And genetically susceptible mosquitoes were almost fully killed by the application. However, those mosquitoes that were homozygous mutant for the Kdr mutation 1016 showed a mean mortality below 25%, indicating a differential selection of the insecticide in favor of resistant mosquitoes. A logistic regression confirmed that, showing that the odds of a resistant mosquito surviving compared to the homozygous susceptible was 0.06, whereas for a heterozygous resistant, the odds was 0.25. In fact, for the 3 pyrethroid-resistant mosquito colonies used, the frequency of the isoleucine allele in the surviving mosquitoes was significantly higher than in the dead mosquitoes, reaching values ranging from 75 to 80%. This, again, is proof that the household aerosolized insecticides are selecting for pyrethroid resistance, and also that removing pyrethroid from public health will be unlikely in reverting pyrethroid susceptibility in the region. In conclusion, Mexico's state-of-the-art monitoring program provides useful information to guide decisions of insecticide procurement and management of insecticide resistance. Insecticide resistance monitoring should be conducted at multiple spatial scales, country to within city, to understand the spatio-temporal scale of selection. Particularly for pyrethroids and Kdr, household insecticides may be critically relevant in explaining the lack of restoration of susceptibility years after they were phased out from vector control programs. I would like to thank all the people and founding agencies that made this research possible, and again, thanks to the organizers for this opportunity. And here's my email if anyone would like more information. Thank you.
Thank you, Dr. Gonzalez, for a thorough investigation. And well, I'm pleased to introduce our next speaker, Dr. Nazni Wasi Ahmad. She works at the Ministry of Health in Malaysia. Welcome, Dr. Nazni.
Hi, thank you, Carolina. Hello, I'm Nazni from the Institute for Medical Research. I'm R. The Medical Entomology Unit is a WHO collaborating center for taxonomy and vectors of public health. And recently, our lab has also been accredited as a GLP lab by the local authority as well as WHO. We are one of the 6 lab in Asia who has been accredited for GLP. Okay, vector-borne diseases is a burden in human population for centuries. And we have in Asia, dengue, Zika, chikungunya, malaria, filariasis. We do not have yellow fever anyway in Asia. That's something good for us. And we have other diseases as well carried by these vectors. And the important mosquitoes in Asia are the Mansonia, Anopheles, Culex, Aedes, and Armigerris. And currently, dengue burden is more prominent than malaria in Asia. As you can see in this slide, which has been published on 7 January 2022, the burden is in India, in the Mekong region, in the Philippines, in Indonesia, as well as in Malaysia and Singapore. So it's very difficult to kill this vector. You need a lot of resources to make sure that this vector is being controlled. The key vector for dengue is Aedes aegypti as well as Aedes albopictus in Asia. And there's another one vector known as Aedes niveus group, which dwells on the canopy and is able to transmit all the 4 dengue serotypes and is responsible for salvitic transmission. Traveling by air is one of the reasons the spread of dengue in Asia. Now or today, you can travel within a day to any of these countries. There's a publication which states that, which has reviewed 59 years of data from 20 countries in Asia, and it has confirmed that dengue serotype 1, 2, and 3 is linked to air travel in Asia. Do you know that mosquito kills more human than man killing man? Okay, Aedes aegypti and Aedes albopictus gained popularity during the Zika outbreak, and WHO has mentioned in one of their teams in 2014, small bite, big threat. The key issues to control dengue is there's no specific antiviral, there's no effective vaccine. Current strategy for prevention is community-based integrated mosquito control. Excuse me. And the most important point is as low as 2 to 3 female Aedes aegypti emerging every day in a locality of 100 people is sufficient for dengue transmission. The overall problem, to control these vectors, we have been using the chemical insecticide, the biological insecticides, and environmental management techniques. However, the overall problem, it remains very challenging. In Asian countries, in most of the Asian countries, if there's a dengue case, thermal fogging and the ULV truck mounted is being used. However, in Singapore, it is preventive, whereby there is a routine fogging taking place. When a single case is reported for dengue, thermal fogging at 200-meter radius is being used. But if there's a dengue outbreak, thermal fogging, truck-mounted ULV using the same insecticides are being used and is being repeated for 7 to 10 days later. Cost estimation of current dengue control measures in Malaysia in a 4 district, which is hotspot, is 22.4 billion, which is equivalent to US 5 million. And for your information, in Malaysia, we have got 100 of districts and we have got 100 of hotspots. This indicates that it's controlling dengue, it's not chicken feed. The DDG of the Ministry of Health said that ideal dengue vector control tool needs to be scientifically proven, affordable, and sustainable. And chemical pesticides has been used for vector control for at least 80 years. This is one of the figures where I'm showing you when the organic compound has been come into the world, which is DDT in 1940, and now in until etopenfrox, which is one of the recent. And I think there is 2 more recent compounds which has been brought into, which is Fludora fusion, and there's one more, I can't remember the name, but there's another compound for vector control. The annual insecticide usage by region is shown in this table, but I would like to bring attention to Southeast Asia, where it's using the huge amount of insecticide. From in the year 2000 to 2009, you can see that organochlorine is the insecticide most used, followed by organophosphate. However, in the year 2010 to 2019, there is a reduction of usage of organochlorine and all the other insecticides. This could be due to the resistance development to some of the active ingredients, and moreover, it could be due to the introduction of other insecticides, alternative insecticides such as the bacterial larvicides, IGR, and spinosins. This table shows you the burden of insecticide usage in terms of diseases. So, malaria has been using the most amount of insecticide, which is 3,000 over metric ton, followed by dengue. But you can see again that in malaria, organochlorine is mainly used. However, in dengue, we do not use organochlorine. And the amount of insecticide used for malaria is 3 times more than insecticide used for dengue control. Insecticides has been used in agricultural pests as well as public health pests, and this has caused the resistance development. Truth hurts, but lies can kill. So under and overdosing, bad spray coverage, extended spraying intervals of pesticides on a large scale of insect vector and agricultural pest control has led to resistant development in public health vectors. Resistance is not restricted to one or the other life stage. A population may become resistant when its LC50 has increased by 10 times. And the larval test is more sensitive than the adult test in detecting changes in susceptibility level, whereby if there is a 10-time increase in LC50 to an insecticide, is equivalent to an increase of 2-fold increase in adults. This is a table which I have taken from Bharti et al. 2021, where she has done a systematic review on the global insecticide resistance and resistance mechanism. But I have selected only those countries which is those countries from Asia, and I have summarized the Aedes aegypti here for a clearer picture. As you can see from this table, there are 14 active ingredients, and India is resistant to 10 of the active ingredients, and Malaysia is resistant to the 10 of the active ingredients. And the active ingredient which is most resistant is Temiphos, which is a larvicide, followed by malathion, permethrin, then comes lambda cyhalothrin. This is for Aedes aegypti, and you can see that even though we have never used DDT for our Aedes aegypti control, but is already naturally resistant. And this is a table for Aedes albopictus, which I have summarized for the Asian countries. And again here, you can see that among the 13 insecticide used, in India, 9 of insecticide out of 13 already being resistant. And similarly in Malaysia, 9 of the AIs are already resistant. Again, Temiphos seems to be the most resistant larvicide in most of the countries, and followed by malathion, permethrin, and lambda cyhalothrin. The resistance, I've shown you that Aedes aegypti and Aedes albopictus is already resistant to most of the insecticides. But we have also done screening on Culex fasciatus, which is already resistant to malathion, penitrathion, and permethrin. Anopheles maculatus, which is a vector for malaria, is still susceptible to most of the insecticides. This is where I am from, Institute for Medical Research. It has been established in 1900, whereby the purpose was to conduct studies on vectors of malaria as well as the diseases, which is malaria. And now the malaria due to Plasmodium falciparum has faded. So the most important disease, the malaria disease now is the simian malaria, which is quite alarming and causing quite a number of deaths. But the institute has also focused its research to controlling dengue vectors. The institute also monitors in the hotspot locality, and monitoring resistance is very important, and insecticides are being used, and that change in insecticide policy are based on sound scientific data. This is a table where I would like to share with you because it has got some interesting results here. It's a knockdown resistance status of field strains Aedes aegypti adults exposed to 0.75% permethrin. I would like to show you that this locality, which I've highlighted here, which is 26.57-fold resistance to Kdr, and this another locality which is nearby 50 kilometers away, and it is 598 folds resistant. The resistance mechanism involved here is due to Kdr mutation, and it could be two different Kdr mutation involved. In IMR, we also evaluate insecticide in an operational setting. If there is a continuous increase in dengue cases in the hotspot locality, even after 2 cycles of adulticidal spraying, MOH will request IMR to evaluate the efficacy of the insecticide used in that area. So what we will do in IMR is that, first of all, we will do the we will use the WHO bioassay testing, just using the lab strain and field strain to see if there is resistance development. Then we will do field evaluation by using cylindrical cages by WHO, and we will put the mosquitoes, the field collected mosquitoes, which is F1 generation, which is very similar to the wild type mosquitoes in these cylindrical cages, and which is being placed in the living room, in the bedroom, and in the kitchens. And you can see these cages here with mosquitoes. So this the operational data shows that resistance has developed to the commercial formulation, and is more evident in ULV spraying than the thermal fogging. It's very clear here. This is thermal fogging. The mortality is around 60% and below. But if you see the ULV spray, you can see that the mortality is below 20%. So you can see that there is a resistant development, because when you see the lab strain here is 100% kill, and this is another strain from rural where there's less exposure, is still almost 100% kill. But in the ULV, but you use the ULV strain, it's still the lab strain has got high mortality compared to the field strain. This indicates that this particular commercial insecticide is already resistant to the field mosquitoes. However, if you see the Aedes albopictus, it's interesting to see that the same insecticide was effective to give a near 100% kill in the field Aedes albopictus. It's very clear here. It's almost 100%, and over here is almost 100%. So therefore, this shows that this particular commercial insecticide is already resistant to Aedes aegypti, but not Aedes albopictus. So what's next? If this happens, what the ministry will do is the ministry will relocate this insecticide to areas where less insecticide are being used, and it will be used for controlling nuisance mosquitoes. And in this area where the resistance has occurred, another kind of insecticide with different mode of mechanism will be used. Resistance surveillance is crucial in providing baseline data for control program and pesticide selection. Early detection of resistance in mosquitoes populations helps in preventing failure in disease control and management. In the IMR, we have developed a rapid resistance detection kit, which we can screen resistance in the field rapidly. We have developed a kit for oxidase, for esterases, as well as for acetylcholinesterase. This kit has been patented in several countries. This is very easy to use this kit because it has got color code, which indicates highly susceptible, moderate resistance, and highly resistant. Same thing is for oxidase and as for acetylcholinesterase. So the test procedure is very simple. You just homogenize insect, which is single mosquitoes, add substrate, add indicator, and read the color code. And by using this in the field, you can very fast, you know whether the percentage of your mosquito population is resistant or is susceptible. This is an example from field collected mosquitoes, where you can see that in a field, the wild type mosquitoes, some of them are highly resistant. There are susceptible mosquitoes in the field, as you can see here and there are also heterozygous resistance, and these are all susceptible mosquitoes. This is for esterases, and for oxidases, you can see that there are highly resistant mosquitoes to oxidase. The mechanism is oxidase, sorry, the mechanism involved is oxidase. And for acetylcholinesterase, If you see, if we use these kits, if it is yellow, it indicates that acetylcholinesterase is present. But if we use, these are 4 replicates, this is another 4 replicates from the same mosquito. These are all single mosquitoes. And when we add propoxo in this wells, when we add propoxo, and if the color still remain yellow, it indicates that a particular insecticide is not sensitive. It's no more. That means acetylcholinesterase is not sensitive. But if it inhibits, that means it's still sensitive. Okay, that means this particular, in this case, propoxo, when we use propoxo, it inhibits the color. So it's become clear. When it's become clear, it shows that the carbamate can still be used to control the mosquitoes. Resistance does not imply that it is impossible to control the resistance population or to prevent disease transmission, or that all population of this species cannot be controlled. Thus, a single report of resistance to an insecticide does not imply that an insecticide is no longer useful either within the local region or globally. I would like to emphasize here that lab data for resistance is not sufficient. You need to complement it with field evaluation in order to conclude and make decision and recommendation of the insecticide in question. With this, I thank you for your kind attention.
Thank you very much, Dr. Nazni. We know it's midnight for you, so we are very grateful for your time. And we are very fortunate today. Our next speaker is Dr. Dina Fonseca from Rutgers University Center for Vector Biology in New Jersey.
I have to let, somebody else has to let go of the screen. Oh, there we go. Okay, it's good. All right, can you hear me okay?
Yep, we can.
And can you see the slides okay? Right size? Is that a yes?
Yes, yes.
Oh, thank you very much, Banu. Okay, well, thank you very, very much for inviting me to participate in this summit. I've just learned a lot, and there's also some really interesting contrasts between a countrywide state-developed insecticide resistance monitoring and what I am about to tell you, what we are experiencing here, specifically in New Jersey. So the title of my talk is Life After Insecticide Resistance Was Detected or Is Detected, and really is a conversation. I developed this talk specifically to talk to the mosquito control programs in New Jersey. Now, it's sort of important, and I'm basically speaking to the converted, but that insecticide resistance can impact control. And, you know, in Malaysia, in Mexico, this may be completely obvious to everybody, but actually it's still often not obvious to some of the constituents that we work with. So there is evidence from publications that in larval stages, insecticide resistance is originally associated with a reduction in the duration of the treatment efficacy that can be seen, for example, as normally you would have 8 weeks of control, and if you have some level of resistance, you may only have 3 weeks of control, and if it gets really bad, you may have very little control or none. And this has been shown in the Caribbean, Brazilian populations. I'll show the references in 1 minute. There's also evidence of both larval and adult resistance that even after 3 rounds of ultra low volume thermal fogging with deltametrin, you saw no actual changes in the numbers of biting adults and also of larvae. And sort of more recently, sort of to do here with in US, we have seen failures of applications of pyrethroids in Florida, sort of implicated in the loss of control for controlling Zika transmission in Miami. So, and again, these slides will be shown, I guess, shared, and so the references are here. I always reference whenever I'm sort of saying anything. Now, working my lab, I'm a basic scientist, or I'm primarily a basic scientist. When I started working on Asian tiger mosquito in 2008, and sort of later on brought in Sébastien Marcom, that was trained in Vincent Corbel in Montpellier on sort of the intricacies of insecticide resistance testing. And it was sort of my introduction to insecticide resistance. Marcom did a fantastic job of looking at insecticide resistance in Aedes albopictus, mostly in the eastern US. He looked at populations from New Jersey, Pennsylvania, and Florida, multiple populations, and he tested 11 different insecticides, including all modes of action, starting with DDT, and both larvicides and adulticides. He also characterized a susceptible strain of Aedes albopictus that is now available from BEI resources, called the ATM New Jersey 95. That was important as a way to create a baseline that we could then compare and evaluate any kind of levels of resistance in field populations. During that study, Sébastien also found incipient and some moderate resistance to both organophosphates, OPs, and pyrethroids in populations in New Jersey and also in Florida. And of interest, he found DDT resistance in Aedes albopictus in Florida. Not associated with a Kdr mutation, but we suspect actually directly associated with a DTTase, a detoxifying enzyme that was specifically associated with DDT. And that was sort of surprising. Albopictus didn't get to Florida until 1986, or somewhere around, maybe 89. And DDT had been outlawed since 1972 in the United States. So this idea that migrating mosquitoes coming in already with insecticide resistance were important. So we've talked about some great presentations on the origin of insecticide resistance. And frankly, a mutation is a relatively unlikely thing to happen. So often, as has been mentioned, you already have a nice sort of brew of more or less resistant mosquitoes in a population, and then you can either drive them to an increase or not. Later on, I had a student, Brian Johnson, that looked at sort of what Sébastien did were bioassays, but Brian was interested in looking at, since we had the tools in Culex pipiens to actually look at the molecular basis of insecticide resistance in a comparison of residential and wetland Culex pipiens here in New Jersey. He found for the first time, not because it was unknown in the US, but it had never been done in New Jersey, the combination of ester 2, A2B2, and also ester B1, upregulated esterases that confer OP resistance in Culex pipiens, and also the classical Kdr mutation L1014 at a relatively high rate, and finding evidence of positive selection, so higher numbers of homozygotes than expected from the numbers of heterozygotes. He also found double mutants of the upregulated esterases and also Kdr mutations, which really becomes a problem when you're trying to do any kind of rotation between the only two class of insecticides available to us for public health of adulticiding in New Jersey, OPs and pyrethroids. So after I done this work, I later on became the director, as I am now, the director of the Rutgers Center for Vector Biology. And this is a program that originally known as the Mosquito Research and Control Program. We do basic research on vectors and pathogens, ecology, evolution, surveillance, and control, so basic science and operational. And we work in close association with New Jersey's 21 county mosquito control programs and also New Jersey residents. We have multiple and regular institutional collaborations with other professional organizations in New Jersey and beyond. And we have, a nice lab. We can do a lot of things. We get a lot of federal and state-funded projects. And because of all of this, I became involved in the WIN, the Worldwide Insecticide Resistance Network, that was, again, started in Montpellier by Vincent Corbel and Jean-Philippe David, and then expanded to 19 countries, and included me as representative of the Center for Vector Biology. This is, again, it was a big learning experience for me, and really trying to think about and what sort of this comparison between statewide approaches and what we have here in the United States, which is literally sort of mom and pop. So it's local development of control, locally funded. And because of that, it can be a very flexible and very powerful approach, but it can also maybe, and this is where the discussion starts, a little bit of hard on the upkeep, on sort of deciding to do something. Now, as part of multiple papers that came out of the WIN, here's a complicated slide that is complicated on purpose. Developing an insecticide resistance management program, it really requires sort of the main pillars of surveillance, research, management, innovation, financial support, and otherwise, including advocacy. That then requires a view of short-term, 1 to 2 year situation analysis, the surveillance, understand what's present in terms of insecticide resistance, and frankly, starting before you see insecticide resistance. So management and rotating the use of insecticides and doing it all this in an enlightened and scientific way. Then you have the actual IRM implementation in 2 to 5 years, and basically a long-term sustaining of the insecticide resistance management. It's a complex process. It requires kind of a satellite view of what's going on. And so where are we? Well, I'm also part, so as a member of the Center for Vector Biology, we're part of the Northeast Vector-Borne Diseases Centers of Excellence. So this is created by CDC. And the NEVBD started in 2019. They did a survey to try to understand how much pesticide use and how much resistance monitoring, insecticide resistance monitoring was happening in the Northeastern US, which is kind of from Maine to Virginia, it's kind of our sphere of influence. The idea was to determine the current extent, determine roadblocks if insecticide resistance monitoring was not being done, and also how NEVBD could help to address this. So the results were basically that was a lot of lack of training and equipment, lack of dedicated funding, time, because you needed more personnel, and in many cases, because again, these are local county mosquito control programs. And as I mentioned, there's 21 counties in New Jersey, sort of equivalent of relatively, so New Jersey is about 9 million people, and it's again separated into about 21 counties. They're pretty high density, and they're locally funded. So the funding, although there's some funding coming from the state, most of the funding is local, and most of them they do control. So finding an area where they can maintain mosquito colonies is kind of a complicated thing to do. So as a response to this, the NEVBD decided to create a centralized pesticide resistance monitoring program. It was initially started by, led by James Bordes, supported by Joe Poggi, Emily Mader is the manager, and Laura Harrington is the director. Since then, now Lindsay Baxter is organizing this. I'm not giving you results about this particular program. I'm showing you some of the results that they have shared at this point and published. So they started this program. It started in 2019. They have data from 2020, 2021, where they basically ask anybody, any program across the Northeast to submit the sample, submit eggs or submit larvae or adults for them to test. And they do that for free. So basically it's just a question of the program sending in the specimens. What they found, in the survey, the primary insecticides used in the Northeastern US, again, from Maine to Virginia, for the larvicides is primarily Bacillus thuringiensis israelensis, BTI, with some level, significant amounts of Lysibacillus sphaericus. In terms of the non-biologicals, the methoprene, so a juvenile hormone mimic, is used extensively, primarily in an urban area, is used for catch basin mosquitoes, for Culex pipiens mosquitoes, and routinely, but not always, again, these are individual programs, they treat thousands of catch basins by applying a briquette of methoprene to be able to control the production of Culex pipiens throughout the season. Small amounts of organophosphates and a few others. For the larvicides, the big bulk of the control is pyrethroids. So 76% of the adulticiding is done with pyrethroid, has been mentioned already, is considered to be least impactful to humans in other, invertebrates. There is some application of malathion OPs, but really the pyrethroids are the primary mode of control for adulticiding. Now, in terms of resistance, there has been detection of moderate to significant resistance to methoprene in New Jersey and Culex pipiens. Not surprising, since, as I just mentioned, methoprene is the primary method of control of these catch basin mosquitoes. It was not found in Aedes albopictus, and I should also mention these are the only 2 species that have been targeted at this point for insecticide resistance testing. And there's also been observation of moderate levels of pyrethroid resistance, including etonfenprox, for both Culex pipiens and in some populations of Aedes albopictus. Okay, so insecticide resistance has been detected in your mosquitoes. Now what? And that's really where I'm coming in. So as a member of the Center for Vector Biology, my job is to support mosquito control programs. And so I want to work with them. I want to help trying to figure out what exactly is the best step forward now that some insecticide resistance has been detected. And I've already mentioned, technically, we should have been doing insecticide resistance management to prevent any insecticide resistance detection. But as also has been discussed, just because you find insecticide resistance in a particular strain collection of mosquitoes that was submitted does not mean that you have widespread insecticide resistance in your population. So according to the CDC, if you find some level of insecticide resistance, if you're still susceptible, do some baseline enzymatic, so P450 esterase testing. Really interesting. Then we'd love to have a discussion with my Malaysian colleague about those rapid assays. If you find some resistance, do some mechanism testing using synergists, and do some field testing. We'll talk about that in a second. And then if you see significant mortality resistance, then start looking at alternative modes of control. So that's basically the idea, but also a lot of discussion about spatial extent, temporal extent, great work, really enjoyed the work from Mexico and understanding this large scale to very local scale development presence of insecticide resistance. So, and then this idea of sort of change control methods. And it is important to mention that reactive alternation is not technically insecticide resistance. And I just want to, I think we all know that, but I just want to point out why this is especially important. The fact is that you can have high levels of resistance and those can be hard to reverse. They can be hard to reverse because the population may not be homozygous. And that has been detected. Populations in Florida and California of Aedes aegypti have been found to be totally homozygous for the Kdr mutation, one or more. And therefore, it doesn't matter if you change your insecticide, it's not going to go away. You may change to OPs, but you're not going to be able to come back to pyrethroids easily. You can have multiple resistance mechanisms. I mentioned we found both Kdr mutations and upregulated esterases in the same mosquito in New Jersey. So rotating these 2 modes of action, it is not going to necessarily do much in terms of changing the increase in insecticide resistance. Of course, as mentioned, presence of additional selection from agriculture and household insecticide use, really good work in Mexico. I really enjoyed that talk. And also the effect of insecticide resistance on vector competence, which has been shown both in Aedes aegypti and thank you, Dr. Wanji, on in Anopheles gambiae. So you can see I've been updating my talk as I was listening to everybody. So it's been fun. So what are the resistance management strategies that I can go and talk to mosquito control programs about? Well, again, rotation, we need to understand residual, we need to understand generation time, and then come up with a strategy to do rotation of insecticides. That is easier said than done. This idea of mosaic may not work for mosquitoes that are like Aedes aegypti or Aedes albopictus, very weak flyers. And so what's the size of a mosaic for a situation like this? And then these mixtures, which we are seeing are already people starting to use, but I'm seeing mostly larvicides with adulticides, which is actually an interesting and definitely something to explore. So we're not seeing, we need to have a situation where there's no cross resistance, the residual, it needs to be the same for the two modes of action. And also it's important to understand the genetic basis. So what I would really like to be able to work with my constituents, I would like a better understanding of insecticide resistant dynamics in our mosquito populations, type and levels of insecticide resistance, which will require these rapid insecticide resistance strategies, markers. And of note, and I really want to talk to my colleague in Malaysia, but we also need methods for non-traditional species. In New Jersey, the big species that we control the most is Aedes sollicitans, the salt marsh mosquito. It can be a vector of Eastern equine encephalitis and is a tremendous nuisance, which is actually the primary driver of control of mosquitoes here in New Jersey. We have hardly any methodology for that species. We're now developing the sort of whole genome sequencing for that species to try to identify some of these, Kdr, esterases, and other markers. We have nothing for it right now. We also have the need for understanding spatial and temporal trends in insecticide resistance. What are the drivers of insecticide resistance, and measures of fitness costs of resistance that we always assume are going to be there. Therefore, when you stop applying insecticides, the mosquitoes are going to revert to susceptible, but are really quite varied, and depends on the species and depends on the insecticide. And again, it depends on if they're homozygote or not. We also need a quantitative measure of insecticide impact on local mosquito control. And that's something that I'm very interested in the next talk by Dr. McAllister, and how we're going to address every single location is different. Insecticide resistance happening at different levels, at different species, for different reasons in different parts of our areas. How are we going to be able to control? What are the strategies for doing this? We need alternative tools for mosquito control, for sure, new actives, and also non-insecticidal approaches that will work in an emergency. And that's really the big one. This is my contact information, and thank you.
Thank you very much, Dr. Fonseca. Okay, guys, stay tuned. I am presenting a new code for quiz number 2. I'm with our agenda for today. Welcome back, everyone. Our next presenter is Dr. Janet McAllister, a medical entomologist at the Arboviral Disease branch of the Division of Vector-Borne Diseases from CDC. Welcome, Dr. McAllister.
Hi, good afternoon, good morning, good evening to everyone. I am just going to double check, and if somebody can tell me you're seeing my screen as
Good on both.
PowerPoint.
Yes.
Yeah.
Okay, perfect. So today I'm going to talk about management for insecticide resistance, and we have had a lot of great speakers throughout the day. There's a lot of points in my talk that have already been touched on by all of the previous speakers, which goes to show that there are some important themes that are coming out in the summit, and that a lot of people are on the same page when it comes to insecticide resistance. So to get started, let me get my here out. Okay, there we go. There are a lot of threats to effective vector control that are out there. There are regulatory actions, whether you can use a particular insecticide or not, what what amounts you can use. There are pesticide attitudes. There are groups of citizens who may have a very bad impression of some of the tools that we use to control mosquitoes. So they may say in a local community, we want you to control our mosquitoes, but you can't use X, Y, and Z. And the local health department or mosquito control program then has to find a way to accomplish that and still interact with and engage with their local communities, and also getting their local communities to buy into their role in vector control and what they can do personally outside of what a government entity might do to help them protect themselves. Their funds are always an issue. You know, there's a lot of great ideas out there, and sometimes there's you know what you need to do, but you just can't do it because you can't afford to do it. And I lump in also incentives for new control insecticides and methods, because it's very expensive for companies to produce new mosquito control products, particularly for vector control, and get a return on that investment. So as a result, the insecticides that we use in mosquito control usually are developed for use in agriculture before we see them being able to be used or registered for use in vector control. And then there's resistance, which can further erode our ability to provide effective vector control, just because the products that we're trying to use may not necessarily be working for us anymore. There are a lot of other threats to vector control, but these are just some of the big ones. And then I want to remind everybody, and this has been touched on by several of the speakers before, is that what actually influences insecticide resistance to develop is not just vector control itself. There are influences on what's being used in agriculture. There are influences in what urban pest control companies might be using when people contract spraying for cockroaches or bugs in their lawn or their gardens. And then what homeowners and businesses may be applying themselves. All of those can influence both the development of resistance in mosquitoes and the underlying mechanisms that develop in a particular population. And also, besides just insecticides, herbicides, fungicides, fertilizers, non-point source pollution runoff, what washes off of the street into the storm drain where larval development occurs, can also influence which direction or which resistance mechanism may be selected in a particular place. And we've also heard that you can have more than one mechanism present in a resistant population. So we've heard about the enzymes, the esterases, the GSTs, oxidases. We've heard about the altered target sites, the Kdr, which is DDT and the pyrethroids, and then the ACE mutation, which affects the target site for the organophosphates and the carbamates. So any one of those alone, any combination of that can be in the mix in your resistant mosquitoes. So it's a very complex problem and issue. And just detecting resistance itself isn't always going to give you a clear answer to what you should do for managing it. So really the first step of managing resistance, it's kind of this iterative cycle where you go out and you do testing and you detect it. Based on that detection, you may or may not take an action to mitigate the resistance or to try and prevent it from developing. And then you evaluate, was that action effective? And at the same time, you're continuing to do this detection, action, and evaluating, and people often don't evaluate changes in their program. They just say, oh, we needed to rotate chemicals. They rotate chemicals, but they don't necessarily evaluate was that actually the right action to take. So we heard there are many tests that are out there for detecting resistance and its underlying mechanisms. Choosing which one is going to be a local decision. The lab testing like bottle bioassay, topical application, WHO assay, these lab-based assays are a great screening tool to use to look for changes in a population, but they don't necessarily mean that your products are not working out in the field. And as we heard before, you really need to follow that up with some sort of product evaluation, some sort of field testing to see, to confirm those results. But they are a great way over time to monitor for trends in your population. If you detect resistance, is it getting worse? Is it staying the same? Are you seeing resistance to things you're not using? Are they getting worse? Are they staying the same? What underlying mechanisms are you seeing? All of those things go into that basic surveillance. Where we have the biggest issue when it comes to insecticide resistance is we have a real data vacuum on what path works when resistance is detected. And I was very encouraged to see a lot of the work that's being done around the world that's trying to address that data vacuum on what choice you make and how it is actually affecting both the control of the vectors and potentially the transmission of the diseases in your part of the world that you're most interested in. So when resistance is detected, you have to decide on an action. And again, that action can be no action, that you detect resistance, but it's not really impacting your overall control because it's a very weak mechanism that's underlying. The resistance is not strong enough that you're seeing products failing in the field. It can need to be a drastic action that, oh my gosh, adulticides aren't working for me anymore. Because as we've seen in many places, there's resistance to both organophosphates and carbamates and maybe pyrethroids all in the same area. And if those are the only three modes of action you have to work with, what do you do then? So with that said, there are some classic areas of resistance management, and these theories have been around for decades. They're nothing new, and hopefully if you're new to insecticide resistance, you're learning something, and if you're very experienced in the realm of insecticide resistance, you've probably heard all of these before, but repetition is always a good thing. Sometimes we tend to forget over time and need to be reminded of things. So I'm going to go through each of these. There is management by moderation, and just like the title of this management strategy, it's using lower doses and using fewer applications so that you're not putting a lot of pressure on your population. It's using chemicals that are short-lived in the environment. And I realize that, you know, we across the globe are dealing with different biologies. We're dealing with Anopheles and Culex and Aedes, but we're also dealing with different diseases and different situations on how we can perform control. So in Africa, where you have rural villages that you really don't have the resources to use short-lived chemicals, using residual sprays, using bed nets so that you only have to visit these villages, you know, maybe once a year or once every 18 months, really precludes you to be able to use short-lived chemicals. Here in the United States, the majority of our mosquito control is done from the middle of a public street using a ULV spray, and those ULV products are formulated to have a very short half-life. Some of our products here have a half-life of only an hour, and then they dissipate from the environment so that we're not affecting other insect species, and we're not we're minimizing the insecticide burden on the environment and on humans. Using local treatments if possible. So they're really the basis of integrated mosquito management or integrated vector management is doing surveillance, looking at your mosquito populations. If you have very low populations and there's no disease transmission going on, perhaps you don't need to be treating as intensively as if you're at the height of a dengue outbreak or some other outbreak. The concept of having refugia, of having areas that are not treated by vector control can be important, because that gives you places where susceptible mosquitoes can reside that can then move their susceptible genes back into areas where you have kind of used your control method and reduced populations. This change in economic threshold is kind of a holdover, because these theories are not just for vector control, they're for agriculture and urban pest control as well. In vector control, we don't really have an economic threshold, whereas for a crop, you can have a certain amount of damage to that crop before you start losing money on it. So that one doesn't really apply to vector control. And then you have the exact opposite. You have management by saturation. So you have these resistance genes out in the population, and you render them functionally recessive by using such a high dosage that that gene, that resistance in the particular individual is still overcome by the insecticide. Incorporating synergist into the product is another way of rendering that resistance gene functionally recessive. This assumes that the genes for resistance are dominant in compounds that you're using, again, have short residual actions, which is not necessarily the case for some of the malaria control and other vector control that needs to be done when you're using wall spraying as a strategy or when you're using nets. And again, for this to work, you need refugia and immigration. And I mentioned that before I get too much further, I do want to mention that these management strategies are really strategies for new chemicals to prevent the development of insecticide resistance. Once you have it, these strategies are perhaps not so useful. And then there's management by multiple attack. That's the third strategy. That's where you use a mix of chemicals. And the idea behind this is that you have two different chemicals with two different modes of action, which makes it much harder for the insect to be able to develop some sort of resistance that is going to work on both of those modes of action at the same time. And then there's the alternation of chemicals using the concept of rotating chemicals or applying them in mosaics. And again, the assumption for this to work, as Dina pointed out in her talk, there's no initial resistance. The resistance mechanisms are different. And they have similar decay rates in the field. So if you mix two chemicals together and one has a very short residual action and one has a very long one that lasts for months, then effectively you're negating your mixture of chemicals, because one of them is out there all the time selecting for insecticide resistance, and the other one is only there for short periods of time. I want to pause a little bit and talk just a little bit about our larvicides. The focus of all of the talks before this one have really been on adulticides, and this talk is also really on adulticides. But when it comes to the larvicides that are available for vector control, almost every larvicide product out there has a different mode of action. BTI works on the midgut. This Lysibacillus spiricus works on the hindgut. And then you have insect growth regulators that are mimicking juvenile hormone, or you can have Timophos, which is working like one of our, well, it's an organophosphate, so it's working on the acetylcholinesterase in the nerve synapse. And so when it comes to larvicides and larvicide resistance, anything, almost anything that you follow on with, if you detect resistance in larvicides, is going to be a different mode of action. And there are products out there that actually combine some of these different larvicides. So again, they're a mixture of chemical with mixture of modes of action. And in the chat right before this started, I gave you a link to some videos on how to do larvicide testing from the Center for Vector Biology in California, Dr. Stephen Su's lab. And I also gave you two DOIs for a couple of papers that Dr. Su published, where he looked at resistance to a particular insecticide, and then he tested other larvicides to see where did the resistance to the methoprene or the spinosad, which he was discussing in the papers, where does it cross to other larvicides with different modes of action, and where does it not cross and give you the opportunity to regain susceptibility. So that's, I think, all I'm going to say about larvicides. We're in a much better place when it comes to larvicides. But I do want to ask the question, out of which of these strategies is best? And is it moderation, is it saturation, or is it multiple attack? And this is where we are really in a data gap. So when it comes to preventing resistance in the first place, there's not a lot of long-term studies that have shown which one is the best to use. And I suspect that at the end of the day, when we get new products, which with different modes of action, especially for the adulticides, that there is some work and some thought put into trying to figure out which one might be the better way to go. So just quickly, we tend to treat everywhere the same, even though selection pressures are not equal. So if you have a city, you have residential areas, you have high density residential areas, you have government areas, you have entertainment areas, you have parks. On the outskirts, you may have agriculture going on. There is not a uniform environment out there. And what we tend to do is to blanket everything using one chemical at one rate everywhere equally, whether there's resistance detected or not. It's much easier to do that, and it can be much cheaper. So your country program could say, this year we're going to buy this product, and everybody's going to use it equally everywhere. What we've heard, and what is true, is that the development of resistance is focal, and the mechanism behind that resistance can be different even to the same chemical. So in some urban areas, you may have resistance develop to say permethrin. I'm going to use that as an example because that's what you're using across the board. In some places, that resistance may be a pretty weak mechanism behind it. In other places, that resistance with the same mechanism may be very strong. So there may be just a little bit of increased oxidases in these two areas. There's a lot of increased oxidases. In other areas, the resistance to that permethrin may be a different target site. It may different mechanism altogether. And then there could even be a third or a combination of those mechanisms behind that resistance. So what influences which way this goes? Like I said at the beginning, agriculture can be influencing what's going on here maybe. What's being done in our parks for recreation and to keep like golf courses pretty and the grass perfect year round can be affecting it. What is happening in industrial areas could be affecting it. So there's a lot of influencers on that resistance to the pyrethroid that can lead to different modes of action. And so our blanket approach of using just one thing across the board everywhere is really not the best strategy. I wish I could tell you, you have permethrin resistance, follow it with chemical X, and then maybe rotate in chemical Y, and it's going to go away. It's never going to be that easy. So the best strategy really is to use data, figure out what those mechanisms are, how bad that resistance is, and then tailor what you do to the area where that insecticide resistance is found based on what you actually found is going on in the area. And this is really a part of integrated mosquito management. This is not something special. You should be doing surveillance for your mosquitoes out there. If you don't have the mosquitoes, you don't have the disease, you don't have a situation where you need to be spraying, maybe you don't need to be spraying. Maybe your response is, this is a high industrial area, and we don't, even though we have resistance there, we don't necessarily need to be doing treatments in that area because people aren't being exposed to our vectors there. So in summary, knowledge and data are going to drive the decisions, whether you rotate chemicals, whether you need to put more emphasis on other control, back off on the adulticiding, do more larviciding, focus more on community cleanup, all of those things that go into integrated mosquito management. All of those are options for you to use. And again, we don't have good, solid information, and every place is going to be unique in what their local problem is. So making a blanket policy on how you're going to respond to insecticide resistance is going to be elusive for us, certainly in the short terms, I should say. And I do want to throw this out here because money is always going to be a factor in the decisions that are made. So if you have areas that don't have resistance, you don't necessarily need to change how you do control. There's no cost difference in what you would be doing with your vector control program. But in areas with resistance, alternating chemicals and using the highest rates, they can affect your bottom line, and they can impact your ability to do mosquito control, because if you're focusing on the resistance and it's taking a lot of your resource in that area, instead of doing that blanket across your entire town or state or country, use those extra resources needed to address that local resistance problem. And then in places where there isn't resistance, having the ability to use your maybe lower cost treatments that you have been using in the past is really the way to go. So I wish that with this talk I could have answered my question that is there a way to have a single answer for all of this. And unfortunately, when it comes to insecticide resistance, there's not a way. And data and knowledge and education are key to being able to address insecticide resistance for us. And I'm gonna leave off there. My contact information, feel free to reach out if you want to email with additional discussion beyond the panel.
Thank you very much, Dr. McAllister. We are very grateful for all your advice, especially for having you live. I'm just gonna ask you to stop sharing.
I'm going to find how to unshare my screen.
No worries. The next person can share and it'll automatically
There.
go off.
There. I stopped sharing, so.
Thank you. Okay, folks, we are going to have a slight change in our agenda as Dr. Lucas is coming ahead. It will be our next presentation, and Dr. Clifton's presentation will be after Dr. Lucas. So with this being said, it is my pleasure to introduce Dr. Kira Lucas, Deputy Executive Director at Collier Mosquito Control District in Florida. Please, the mic is yours.
Can you guys see my screen? Oh, there we go.
Yep, we can.
Okay. So good afternoon. I'm Dr. Kira Lucas, Deputy Director at Collier Mosquito Control District. And I want to start off with thanking Banu, Leanne, and Carolina for arranging this Resistance Management Summit and inviting me to participate. Today I'm going to discuss the insecticide resistance status of Collier or container inhabiting mosquito species in Collier County, Florida, how we do our monitoring, and as well as some of the insight into how we currently manage those mosquito populations. So for those of you who don't know where Collier is, we are located in Southwest Florida, about 90 miles from Miami on the Gulf Coast. As more as 70% of our county consists of environmentally sensitive public lands, we currently cover about 400 square miles in the areas of development that's shown in green. In Collier, we have more than 50 species of mosquito, and the district targets both disease vectoring and pestiferous mosquitoes. We practice an integrated mosquito management approach where we actually tailor how and when we treat to the specific mosquito that we're targeting. So how we target Aedes tenurinchus or Mansonia, very different than how we target Aedes aegypti or Culex pincafaciatus. We do perform resistance testing on all of our highly targeted species, including our nuisance species, such as Aedes tenurinchus. Luckily, we have yet to identify resistance in Aedes tenurinchus due to their migratory nature, but I do know that other mosquito control districts in Florida do struggle with resistance in Aedes tenurinchus. Today, actually, I'm going to focus on our container inhabiting mosquito species, and that's going to include Aedes aegypti and Culex pincafaciatus, and the resistance status of those particular species. These mosquitoes often produce in neighborhoods in our county, such as the one pictured here, often in areas that are not deed restricted or governed by homeowners associations. And as you can see in these images, there are lots of containers around the homes and other potential sites where these container breeders can be producing. Our surveillance for container inhabiting mosquitoes actually started in 2016. Our understanding of the dynamics of these species weren't well defined until the county was threatened by potential Zika virus transmission. And then subsequently, the state of Florida was placed under a public health emergency due to imported and local Zika virus infections. Unfortunately, before then, we didn't know where these species were or where they could be found. And our leadership at the time had to start an Aedes surveillance program from scratch, and it was very reactive. But since 2016, we now have a very clear understanding of where our container inhabiting mosquito species are most abundant. So this is our Aedes aegypti, and this is our Aedes albopictus. And one thing to note is that these Aedes mosquitoes, Aedes aegypti and Aedes albopictus, account for less than 1% of our district's mosquito collections. But because of their importance as disease vectors and our subtropical weather that supports production year-round, the majority of our understanding of resistance and the research into developing new strategies for their control is extremely important for the district, and we do put a lot of effort into evaluating new methods to control them. The same goes for Culex quinquefasciatus, and we know where those are located throughout the county. These mosquitoes are important for us, and they count for nearly half of our West Nile virus mosquito pools. And we will target our treatments for them based off of where the hotspots are for West Nile virus. During the same time as the district started to become threatened from Zika virus, we were also working to incorporate pyrethroid-based materials for adulticiding. Our district exclusively used dibrom operationally before that, and then Baytex even before that. What we didn't know in 2016 was that both Aedes aegypti and Culex quinquefasciatus mosquitoes are resistant to pyrethroid-based control materials. And the district and our collaborators have been really good at documenting this resistance. And more recently, we've identified nailid resistance in our Aedes aegypti in the county. And we do not yet know how widespread this resistance is, and we're currently working on understanding the resistance countywide. So I'm going to go through how we perform resistance testing at a district level. You guys have heard about the majority of these methods today. Our main method for detecting resistance is the CDC bottle bioassay, using both technical grade and formulated products. We always look at both. After we identify potential resistance through our bottle bioassays, we do look at the factors that can contribute to resistance. Our district does have a real-time PCR machine that we use for disease testing. We can also use that to perform Kdr genotyping. So we're able to have nice data on the status of Kdr, looking at different populations throughout the district, and I'll go through an example of that today. And just because your mosquitoes carry a Kdr mutation, it doesn't necessarily mean that it contributes to your resistance issues. So we do phenotyping assays for Kdr. And then we're also able to do metabolic resistance assays through the use of inhibitors. And I do want to note that I'm not sure if they do it anymore, but the CDC was offering kits to mosquito control districts where they provide everything you need to do this work besides the Kdr genotyping, and is readily available for districts. And it's been a really good resource for us. So in terms of Kdr phenotyping, that's also known as a recovery assay. It's very similar to a CDC bottle bioassay. Actually, you just do a CDC bottle bioassay, but after the 2 hours are up, or your 120 minutes are up, you can transfer your mosquitoes into holding cages, and then you look for recovery after 24 hours later. So if you see that mosquitoes are dead at 120 minutes after your bottle bioassay, but you come back 24 hours later and they're back and flying around again, then that means that you might have resistance attributed to Kdr. For our metabolic assays, we use a modified CDC bottle bioassay where mosquitoes are exposed to an inhibitor before the bottle bioassay is performed. So you will expose the mosquitoes to an inhibitor for 1 hour, let them rest, and then go and just do your regular CDC bottle bioassay. And so when you compare a regular bottle bioassay to ones that are exposed with inhibitor, it'll provide you information on the metabolic mechanism. Reduction in the resistance phenotype with exposure to the inhibitor will indicate resistance attributed to the specific metabolic mechanisms. And so those inhibitors include PBO, which looks at oxidase metabolism, DEF, which looks at esterase metabolism, and DEM, which looks at glutathione transferases. And then as Janet said, and others before her said, just because you identify resistance in the lab, that does not mean you necessarily have resistance in the field to formulated products. So we do take our work to field cage trials. We do ground trials using a hand fogger or a truck mounted fogger that's readily accessible to most districts. And then for us, we have an aerial program, so we can use our, usually we use our rotary wing aircraft for field trials. And then again, I want to point out that everything that I just described here is very straightforward and easily attainable for mosquito control districts as long as you have the staff to do it. Like I said, the CDC can provide, used to at least, provide a kit to do a lot of these assays. And then I do want to point out a lot of labels for pyrethroid-based materials specify a requirement for resistance monitoring. So I always highly suggest that if you are using a material that does require that you should have a resistance monitoring program in place. So as mentioned, our Aedes aegypti are resistant to both pyrethroid-based materials. This is pyrethrum, and then a sumethrin, and we see resistance to our Collier in our Collier strain. Our Collier strain is the one with the circles here. And then as well as to Nalid. We have not identified resistance in malathion, but we do see it with Nalid. And so you can see here, we have resistance in our Collier strain. We have shown the resistance to pyrethroids is attributed to Kdr mutations, resulting in the conversion of phenylalanine to cysteine at codon 1034 and valine to isoleucine at codon 1016 of the voltage-gated sodium channel. We also know that this resistance is attributed to esterase metabolism out of work from Eva Buckner's lab. And we have done field trials. They're not published, but we have done plenty of field trials, and we know that resistance to our pyrethroid-based materials that we use at the district is common in almost every Aedes aegypti population that we have tested. In terms of our Culex fasciatus, we see resistance to both pyrethroid-based materials. Again, here's testing pyrethrum. You can see our Collier population and sumethrin, and here's our Collier population here. But luckily, we have not identified any Nalid resistance in these in this species. So today, I'm just going to go through some of the work we've done in more detail on Culex fasciatus, mainly because this work was performed entirely by our district and is the most thorough for all the different locations that we've looked at. So to investigate Culex fasciatus and produce a more thorough analysis on the resistance in our county, we collected mosquitoes from several location sites throughout the county. So these areas are much more populated than these areas out here. These are more rural areas. And that is one important thing to kind of think about as we go through this. We started off with Kdr, and that was mainly because we knew there was resistance in our Culex populations from some of our bottle bioassays, and we already had performed Kdr genotyping in Aedes aegypti, and so we were curious to see if this type of resistance was also found in Culex fasciatus. That's kind of where our curiosity was. That's the way we went with this project. We assayed for the L1014F mutation using a TACMAN assay. And as you can see, remember Cambria Park, Sugden Park, Naples Manor, these areas here were much more urban. We saw a much higher frequency of this mutation in those populations. And then as we went out to the more rural areas of the county, we saw much less a lower frequency, or not even detected, or detected at a very minimal level. So the mutation was found in highest frequencies in areas that were more urban, while decreasing in frequencies in areas that were less populated. Our question was whether this mutation can be used as an indicator for resistance and if it had any operational significance. So we took the population with the highest frequency and the lowest frequency. So we looked at Cambier Park, which was the highest frequency, and Sable Palm, which was the lowest frequency, and subjected them to several bottle bioassays. Our results from that indicated that the population with the highest frequency for the mutation, which was Cambier Park, displayed a higher level of resistance than the population with the lowest frequency. Cambier Park is the solid line, and Sable Palm is the dashed line. So higher frequency, Cambier Park, lower frequency, Sable Palm, and we saw the resistance different. But what we did see with both populations is that they did display some level of resistance to these materials, and that was both testing pyrethrum as well as sumithrin. We also looked at formulated product. We saw no resistance with nalin. So we want to determine which factors were contributing to these resistances so that a proper adulticide could be chosen to target these species, especially since we were trying to incorporate pyrethroid-based materials into our program. So we first performed the Kdr phenotyping through recovery assays. When the mosquitoes from the population with the highest frequency of the mutation were exposed to the material, we saw a decrease in mortality 24 hours post-exposure. So this will show you at the 120 minutes, sorry, and then the 24 hours post the bottle bioassay, and you see that there was a recovery. Here we had around like 40% dead, and then over here we had more like 20% dead. So there's a large rate of recovery. And that was true for both pyrethrum and sumithrin. And so that's indicative that some of our resistance was in fact due to the Kdr mutation for at least Cambier Park, not so much for Sable Palm. And then of course, we looked at the frequency of our allele of our Kdr mutation and how it related to recovery. How higher the frequency of the mutation, the more recovery we got. We also performed the modified CDC bottle bioassay to look at metabolic resistance. We exposed both populations to inhibitors. Those inhibitors included PBO, which looked at oxidase metabolism, DEF, which looks at your esterase, and DEM, which looks at your glutathione transferases. And so with both Cambier Park as well as Sable Palm, we saw an increase in mortality when we added the inhibitors PBO and DEF, which indicates that both esterase and oxidase metabolism contributes to the majority of our resistance in our Culex mosquitoes. And then lastly, we wanted to take this, we wanted to know if this resistance in the lab assays actually attributes to resistance in the field. So we set up field cage trials, and indeed we did see a reduced efficacy in the products in our population with the highest frequency of the mutation, or with Cambier Park. Cambier Park is here. We definitely saw resistance to, or a reduced efficacy to Maris 2.0. That's how long ago we did this work, and Bill 1010. And so both of these products, this use pyrethrins, this uses sumithrin. Sable Palm, we actually were able to get a pretty decent kill, and so we were happy with that. But we, from an operational perspective, we came to the conclusion that pyrethroids can be used against Culex faciatus depending on which area of the county we're targeting our mosquitoes. So if there are more urban areas where Kdr Park is, then we're probably going to need to use an organophosphate to target those mosquitoes for adult control. But if we're looking at more rural areas such as Sable Palm, we can use a pyrethroid-based material to provide proper adult control. Currently, we use Dibrom, which has Nailed, and Duet HD, which uses Sumethrin and PBO to target Culex faciatus, depending on where in the county they are. But you know, the question is adulticide always the answer, and can it be effective for our Aedes aegypti populations that show resistance to pyrethroids and Nailed? And then how can we manage this? So this is what we used to do. We used to strictly apply Dibrom to target mosquitoes. It was really our only method of targeting mosquitoes, and we were mostly targeting nuisance species. In fact, there was one year where we applied more than 14,000 gallons of Dibrom. And of course, we did change how we delivered the material to be more cost effective and ensure that we were reaching our target, but it was still adulticiding, and we were heavily adulticiding with no other methods for control. And then in the past 6 years, we've really changed how we target mosquitoes, especially those that are exhibiting resistance to currently available adulticides. We now use an integrated mosquito management approach where we tailor our control methods depending on resistance status and the specific vulnerabilities of the targeted mosquito. And you can actually see that as we have added more and more larvicide and other control methods, our adulticide efforts have decreased. And this is just another way of showing that as we increase our larviciding, we're actually seeing a reduction in the adulticide that we use. I think I'm getting close to running out of time, but in terms of container inhabiting mosquitoes, in 2016, we did adopt the use of liquid larvicide for wide area larviciding, which is currently our only means of control for Aedes aegypti. In 2016, we started with a buffalo turbine and a liquid larvicide unit with a district helicopter. That was mainly in response to Zika, but we've expanded our capabilities since then. We're most experienced with Vector-Bac WDG applied at a rate of 0.5 pound per acre. And since I'm running out of time, I'm going to go ahead and just race through this. But we usually see about 2 to 3 weeks of control. And that is true with both of our truck, which I showed in the last slide, as well as our drone-based applications for WDG. We usually see control for larvae for about 2 weeks, and then we see control for the adult mosquitoes for about 3 weeks. So we like to make sure that we're hitting our hot spots for Aedes aegypti every 2 weeks. And this slide really summarizes our management program when it comes to container inhabiting mosquito species, using a combination of research and surveillance, extensive resistance testing, education outreach, biological control, wide area larviciding through our MISTers and UAS applications. We're able to effectively devise a strategy to combat our container inhabiting mosquito species. And with that, I'm going to leave up this slide with my email and then answer any questions during our panel discussion.
Thank you so much, Kira. That was great content. I'm just going to ask you to stop sharing. Thank you.
Thank you.
Well, we are heading to the very interesting, the last presentation. I'm sure you will all be sad because the content has been so amazing. I am pleased to introduce our esteemed colleague, Dr. Mark Clifton. He's the Executive Director of the North Shore Mosquito Abatement District, just outside Chicago, Illinois. Welcome, Mark.
Hello, hear me and see the slides okay?
We can hear you. Not the slides.
Yeah. The slides are loading. Okay.
Good now?
There you go. Yeah, now.
Yeah.
All righty. So thank you all for having me. My name is Mark Clifton, and I'm the Executive Director of the North Shore Mosquito Abatement District. Just north of the city of Chicago, there's an aerial shot looking south across our district towards Chicago. And today I'm going to talk about another kind of interesting aspect to resistance that I think sometimes is overlooked, as well as its implications for viral transmission and ULV control. And that is really that blood-fed mosquitoes exhibit a kind of transient tolerance to pyrethroid materials. So let me get started here. So there's studies that have looked at the impact of natural kind of existing populations are rare. There's really not a ton of them. There's a lot of studies that look at the effect of an insecticide on an insect in a laboratory. There's a variety of studies that look at cage field trials, especially in our field. But when you start getting out towards an impact of ULV or a treatment on a whole natural existing population, or even the impact on disease or disease transmission, those studies are much more difficult, and so they're much rarer. And so this whole line of research really started because we wanted to understand what our ground-based ULV approach was doing to a natural population. We've all seen cage field trials. We know how things work in the lab, but we wanted to know, how is this going to work out in the real world, in the wild? And the first question that we really had to ask ourselves is, what makes up a population of mosquitoes? Well, in our district, we're about 70 square miles, so rather compact. We have 13 different communities, and you could think of a population maybe as all the mosquitoes that exist within our district. That could be our population. We could divide this population further because we do have two main vector species for West Nile virus. That's Culex restruens and Culex pipiens. So that maybe could be our population. But you could also divide your population even finer. Your entire Culex population is actually a mixture of mosquitoes of all different ages and all different physiological statuses. There are blood-fed Culex, ones that have just fed on blood either 24 or 48 hours ago. You have gravid Culex who are looking for a place to maybe deposit eggs. They've digested their blood meal. You have host seeking, either they're newly emerged or they've laid eggs and they're looking for another host. There's also male mosquitoes and possibly other kinds of physiological state of mosquitoes out there. And there's a variety of different trapping mechanisms for each of these subtypes or subpopulations of mosquitoes: resting traps, gravid baited traps like we use here, CDC gravid traps, or maybe host baited traps with CO2. And so for us to observe a natural population during and after a ULV treatment, we decided to set up stations that contained a modified gravid trap that contains a BG counter, and then the standard BG sentinel with a BG counter. And the idea here was to collect really fine resolution data before, during, and after a ULV treatment. And if you're not familiar with the BG counter, it essentially can count mosquitoes and relay that data wirelessly every 15 minutes. So it provides a really sensitive view on what's happening with the ULV treatment. So we did a series of treatments in 2018 in Skokie, Illinois, which is a kind of a dense suburban urban area in our treatment maps C11 and C18. We did a variety of treatments throughout July during the height of our viral prevalence and kind of West Nile transmission season, the end of July and into August. And we use Duet, which is a synergized pyrethroid, at about the maximum label rate, just under the maximum, by truck with a Clark ULV Clark Cougar. And then we monitor our BG counters continuously during that time to see what was going on. And so as you would expect, you do see a reduction when we apply the material by ground-based ULV. In dark gray is our host-seeking mosquitoes, and they were reduced. And then a few days later, you'd see a rebound after that reduction. But what was interesting is with gravid mosquitoes, we didn't really see the same reduction. In one case, there was a reduction. In another case, the population kind of went up. And in another case, the population just kind of moved sideways. So we started seeing that our host seeking and our gravid mosquitoes were behaving differently to this ULV treatment. We expected them to be identical, and that really wasn't the case. In C11, we didn't look at gravids, but just host-seeking mosquitoes. And again, we saw a pretty clear indication that ULV reduces the population when it's done, and then followed by, a rebound sometime thereafter. Just to visualize the difference in effect between gravid mosquitoes and host-seeking, we used a bivariate analysis, and we put host-seeking female mosquitoes on the x-axis and gravid mosquitoes on the y-axis. And then we connected with arrows the day before to the day after treatment. And so this way you can visualize the effect of treatment on both populations. The fact that the arrows move strongly to the left indicates host-seeking mosquitoes were being controlled, but the fact that the arrows don't move strongly down indicates that gravid mosquitoes were not really being controlled the same way. We really would have expected a similar result. And in one case, the population of gravid mosquitoes actually went up over after treatment. So really inconsistent results. To put numbers to this, we basically over 5 treatments found about a 65% reduction before when you compare after treatment to before. And this was really pretty regular. There were 63, 69, 63, 68. So really about 65% pretty consistently. If you look at gravid mosquitoes below that, you see that they're only controlled about 30% mortality or a change of 30%. And that was really irregular, everywhere from minus 68% to increases of 23%. You see in host-seeking mosquitoes, we had some large rebounds in population after the treatment. And then host-seeking mosquitoes, you just didn't see that same pattern. So these two kind of subpopulations were definitely behaving differently. What was most concerning is we really expected better control at that label rate in our host, at least our host-seeking mosquitoes. We expected, something upwards of 90%, we were hoping, and we only saw a really transient 65%. So this really got us curious about what was going on. And we had no information about resistance in this district that had ever been produced prior to this. So we went back to the basics with the CDC bottle bioassay. Probably should have done this first before trialing, spending all summer doing field trials, but sometimes you just take the route as it unfolds. We've done hundreds of bottles in the last 4 years looking at sumethrin, permethrin, and malathion, and we see averaged across the whole district only about a 74% of mortality with sumethrin and about 59% with permethrin. And then parallel work by Chris Stone at the Illinois Natural History Survey Medical Entomology Laboratory has found this as a common pattern throughout the state of Illinois. Permethrin and malathion resistance can be found just about everywhere. We also went back to the cage field trial to try to understand what a susceptible versus our North Shore resistant mosquito would look like. And so we ran a trial side by side. And again, you see, when we use Duet at that kind of upper label against susceptible mosquitoes, we got fairly, acceptable control. And then against our North Shore wild type resistant mosquitoes, again, 53%. So all the lines of evidence are kind of converging to the same general trend. You have 74% mortality in the bottle bioassay, at 53% in a cage field trial, and 65% mortality in a natural population. So there's a substantial level of resistance that exists in our Culex pipiens populations. Our Culex restruans seem completely susceptible to all of these same materials. When I don't have the data here, but they don't show the same pattern whatsoever. Now, going back to the gravid mosquitoes, this was the next part of the puzzle that we really wanted to understand. Why are these only half as well controlled as their host-seeking counterparts? And half the time, the answers can be found deep in the literature. So going back to 1987, we find that this has been addressed in the literature in the past, and it has implications for what is happening today. 1987, fenvalerate and permethrin, the LD50 had to be, took basically twice the amount of material to kill Culex pipiens 24 hours after a blood meal. The LD50 was nearly double for both of those non-synergized single AI materials. And if you look at in 1990, a similar trend was found with Culex pipiens and synergized resmethrin, where it basically double the amount of material was required at 48 hours after a blood meal. So what we're looking at here is you need basically twice the amount of material to kill mosquitoes, or Culex mosquitoes at least, somewhere 24 to 48 hours after a blood meal. Banu did some work with the Valent's formulation as well as with Duet and a wind tunnel to kind of re-explore these prior results, and found that with susceptible mosquitoes, we didn't see, or he didn't see in the wind tunnel experiments very much of a change after blood feeding. But when you use resistant mosquitoes, you again see the same pattern at 48 hours after a blood meal. You have 65% mortality in non-blood fed, and then you have, that's right over here, and then you have 35% mortality in blood fed. Again, about half of the mortality once they're blood fed. It's interesting, the Valent product work or formulation worked really well, even with blood fed mosquitoes. Even if they're resistant, you remained upwards of 90% mortality. So that's really promising. So just to summarize some of the results we've been seeing, we have gravid mosquitoes exhibit about half mortality when they're resistant as compared to host-seeking mosquitoes. So in the wind tunnel experiment, we had 66% mortality and then down to 35% after a blood meal. Okay, so what does this all mean? Well, it means that mosquitoes are not all just one big fungible population. Every day, a new cohort of mosquitoes is emerging, and each cohort is different than the cohort that came the day before. An individual mosquito, and this is simulated data to show the pattern, but an individual mosquito is really cycling through various levels of susceptibility. They spend about half of their life with far reduced susceptibility because they've blood fed, and about half their life they're host seeking and they're more susceptible. And then you add in all the other kinds of tolerances that we learned about today, metabolic and Kdr, and their total mortality just really drops, or the mortality that's available really drops. And so there's a way of trying to understand what's going on if you want to apply a ULV treatment to a population. And this is a model that was adapted from Chester Moore's work in 1990 that I updated for Excel to try and understand what does it mean when you look at 100 mosquitoes and they're all of different ages and they're all of different physiological statuses and they're all of different resistance statuses. What's going to happen with that treatment? And so in this model, which I think is a great conceptual exercise, and remember, this is all simulated data, but it helps us keep track of what's going on with this highly complex, highly structured population. We have a random number of mosquitoes that are entering the model I put it between 75 and 125 enter the model every day. The model progresses from left to right. So days in the model go 1, 2, 3, 4 as the model progresses. And the age of the mosquito that enter the model progress as you move down. So this way you're matching, you have a kind of an encyclopedia, say on day 10, of all the mosquitoes that exist and all of their different ages and gonotrophic states. Mosquitoes move diagonally through the model, and they suffer like a 5% daily mortality. So they have a 95% daily survival probability every day. So there's a natural attrition that occurs to the population as they move down these steps in the model. So if you have 119 enter on day 1, you'll have 113 and 102 and so on and so forth. And then by the time you get out to like the third gonotrophic cycle, there's not that many mosquitoes, but those are the most important mosquitoes because they're the ones most likely to have a disseminated infection. Those are the ones that are going to infect humans or infect birds. And so those are really the key if you're looking at a public health intervention, is those older mosquitoes. So we can assess the effect of a ULV treatment by just assigning a mortality to each of these different life stages or gonotrophic stages. So imagine we have a material that has a 95% mortality for all stages. And there are materials that are probably pretty close to this that are non-discriminating when it comes to gonotrophic state. And what you see is a really broad effect on the age structure of the population. You don't see any older mosquitoes, and they don't exist for around 10 days. All different gonotrophic states are well controlled. Blood fed 24 hours, blood fed 48 hours. They're all just kind of wiped out. You see the biggest reductions in your population, of course, in your newly emerged, because they're the most abundant part of your population. I want to contrast this with the kind of treatment effect that we're seeing here in the North Shore. We have, in host-seeking mosquitoes, about a 65% mortality. If they're blood-fed 24 hours, I picked the midway. This is a conceptual exercise, about 47%. Blood-fed 48 hours, about a 29% mortality. And then their susceptibility returns at 72 hours after blood feeding. And what you see here is broad swaths of the population are able to persist, especially in your blood-fed 24 hours and 48 hours in their first gonotrophic cycle. They kind of produce a wave that persists out into that older mosquito cohort, days 23 or 24 in the model, 2-week-old mosquitoes. You see that there, and you see it in mosquitoes in their third gonotrophic cycle as well. And then all your other mosquitoes are just poorly controlled. So if you're looking at your oldest mosquitoes, they're still there. The effect on the age structure was not nearly so profound. So I just want to highlight again what these two different scenarios look like. The top is 95% control through all life stages, and the bottom is a variable control coupled with some level of resistance. And you can see, and if you look at your potentially infected mosquitoes, and this is in the red squares, potentially infected mosquitoes are any mosquito from basically day 3 on. They've blood fed and they're potentially infected. And you see when you have 95% control, you get a tremendous reduction down to just 18 mosquitoes, and then a slow buildup back to the model's steady state. If you're dealing with resistance, they're barely impacted. You still see 2/3 of the mosquitoes still exist that are potentially infected with West Nile. We can look at this a little finer, maybe more graphically. This is the same data, but just displayed on a typical line graph. And your susceptible mosquitoes, again, are well controlled if your treatment is on day 14, and then you see a rebuilding of the population. Resistant mosquitoes seem to be not so poorly controlled. It seems like it's almost as good. The problem here is that difference between susceptible and resistance is almost completely made up of newly emerged uninfected mosquitoes, not the part of the population that's the most important if you're looking at a public health intervention. We can look at this another way by only identifying those mosquitoes that are older than 11 days. And remember, these are the mosquitoes that have a disseminated West Nile infection. They're the ones perpetuating this infection between birds and mosquitoes, or maybe infecting people or horses. And if you look at just that cohort, those oldest groups, you see in your susceptible mosquitoes are nearly eliminated for up to 10 days. In your resistant mosquitoes, you still have a fair amount of them. 5 out of approximately 100 are still there. And you can see this wave pattern because those are the resistant gonotrophic, those are the gravid resistant mosquitoes perpetuating out further into the model, creating this kind of bouncing that is occurring. So, where old and susceptible mosquitoes are nearly eliminated, your resistant mosquitoes, they really persist and they remain a threat, and they allow that to continue on. So, just in conclusion, we have widespread resistance to synergized pyrethroid products in our district, and then probably also across the state of Illinois. Mosquitoes after a blood meal develop a further resistance that really affects the age structure of the population. A reduction in treatment effectiveness is only one dimension of resistance, and that's the dimension that we usually come to understand, that we see some reduction in mortality. But there's a deeper kind of third dimension if you think about the age structure of a population, and that each cohort is different as an effect and is affected differently by ULV, and it's also differently affected by its risk of carrying West Nile virus. If you're only looking at total treatment effect, then the age structure doesn't matter. Total abundance is fine. You can just look at that. But if you're looking at a public health impact and what what treatment is likely to really alter the trajectory of viral transmission, then the age structure of the population becomes a lot more important, and resistance really has a big impact, at least conceptually, just with a simple model on how that would look. So a modest reduction in control effectiveness may seem like it's not a huge deal, but it actually is a lot more serious because of the failure to strongly impact the age structure of a population. So I just wanted to highlight this kind of other dimension to resistance that I think is pretty important when we think about, oh, we have a 20% reduction in mortality. Does that really matter? Can't we just use more material, or can we just do it more often? Yeah, it does matter if you're looking at a public health intervention where you're trying to reduce the risk of illness in human populations. So with that, I just want to thank the Midwest Center of Excellence. They've been really supportive and helped us do a lot of this work through having interns or fellows or graduate students come and work with us. I want to thank some of our staff, Chris, who's our laboratory director, and then Roger Nashi, who helped design and analyze a lot of these experiments as we did them. And then Justin Harbison at Loyola, who also helped us conduct a lot of these experiments. And then Valent for being such a great collaborator with all of this. So thank you so much.
Thank you, Dr. Clifton. That was amazing. I want to go back in time and be your intern.
Thank you.
Okay, folks, we are now in the open discussion, questions from the audience. So please, let's just, Leanne, you have some questions already?
Yep.
Well, you can enter new questions if you need to.
Yep. All right. We're going to do our best to get through all these questions. We're a little limited on time. So if for some reason we don't get to your question, we will go ahead and push these to the presenters and have them follow up with you later, or we'll make sure that you get these answers that you look for. So I'm going to do my best to direct these to all of the right people as they were coming in. I do want to make a couple notes. If you haven't checked out the chat, there were a couple questions that went in there and were answered. I did see a couple of questions that Nick answered about the topical assays and if there was protocols and publications regarding that. And Nick just did mention Mr. Aldridge, I believe, does have a publication out there. So check the chat. There's a couple questions flowing in there as well. If you have new questions, please add them in the Q&A. All right, let's get started. So I'm just going to kind of start from the top here from what I have. This looks like a CDC question. Since the CDC dropped mosquito net, where they were collecting resistance data, where can this data be reported now?
So I'll jump in.
Please.
I don't know. We dropped the reporting for mosquito net because in that system, districts had to report to their state health department, which would then report the data, and we were getting very few records. So we felt that it needed to be revisited. We are revisiting that. We are internally discussing how we can get a system in place that bypasses state health departments and goes directly to mosquito control programs so they can put their data in. We are very supportive of the Vector-Serve, which a lot of states are adopting, which does allow them to put their own resistance data in to share within their state. But we don't have an answer yet.
All right. Thank you so much, Janet. And also,
I was going to say, if I could add to that, I was going to again say, like, Vector-Serve is great. If your state's using Vector-Serve, I totally recommend putting that. If you're in the US, one other area that I can recommend is publishing that data. And I know I'm asking you to do something when you've already got limited time, but the lowest threshold to actually get that done, I'd recommend putting it into the Arthropod Management Tests Journal. Dr. Michelle Brown from West Valley is the subject editor for that. There's two sections, one for lab bioassays and one for field medical and veterinary work. $250 for the publication, and it's open access. So I know that I'm asking for a little bit more, but she's fantastic and would love to help you get that published and put in there.
Can you say the name of the journal again, please?
Sure, I'll put it in the chat as well. It's the Arthropod Management Tests.
Awesome, thank you.
Great, thank you for that. And as I see in the chat, and I want to pull it up too, I noticed that Janet and a few other of our CDC counterparts have put in the chat a website site for you to get the CDC's bottle bioassay kits, both in the US and outside of the US. So check the chats there. I saw them pop up a couple of times if you're interested in that information. All right, I'm going to move on to the next question. I think this one might be for our counterpart in Mexico. Let's see. What product is being used when the Aedes aegypti population since 2016? What product was used to phase out the pyrethroids in Mexico after that time?
Oh, I'm Gonzalo Vasquez. So Gabriela couldn't be here, so I'm filling in. I'll try to comment as well as possible. So the main thing is they moved to organophosphates, and chlorpyrifos was one of them. Cielo, I think, is one of the commercial names for the other one that they're considering now. I think they're moving away and using products that primarily rely on organophosphates. Malathion is the other one. And that has been the primary type of insecticide for adulticides.
Great. Thank you so much for that. I think this one is for Jennifer Henke. Is there any agency or college collaboration with you in Puerto Rico?
So the work that I was presenting was what's published and what's been shared in that database. So I'm not directly working with anybody in Puerto Rico. Obviously, Dr. Grayson Brown and his team have been doing all sorts of work. If you're looking for a connection, I would recommend that Puerto Rico reach out to be part of the Southeastern Center of Excellence. I'm sure the folks there would be happy to try to pull people together for any work that's being done on insecticide resistance.
All right, awesome. Thank you for that. All right, my next question, I believe, is for Dr. Nazni here. Is it possible that resistance mosquitoes from other countries, such as India, being imported into Malaysia via air travel or non-fumigated shipping containers and breed with local Malaysian mosquitoes, and worst, come out worse, becoming dominant mosquitoes in the population in Malaysia?
I think, thanks, Anil. I think it's possible for imported mosquitoes to be colonized, let's say in Malaysia, if they come in. But I do not have data for that. But it's possible, I think.
Dr. Nazi, I think your microphone might be going out a little bit because we can't hear you too well.
I think it's possible for the mosquitoes, let's say from resistance locality, to come into Malaysia and be colonized. But at the moment, I do not have data for that.
Okay,
the
great.
possibility
Thank
is
you.
there. Yeah.
Okay. All right, great. We did have one more question for you while you're here. Where can we go to get the rapid insect resistance test kits that you mentioned and developed?
There is a company who is commercializing it in Malaysia, but so far it is being sold in our local market. I mean, local. It's a small. It's not many people are using it, so it's more to vector control people who are using it. It's basically in Malaysia. But if anybody wants, I can always share the company's name and address.
It would be
Okay,
very
great.
interesting.
If somebody wants it, we'll.
Yeah, it would be
Yeah, we'll
interesting
make
to
it
check on different populations with different genetic backgrounds, like in the lab, known backgrounds. That I'll be following up. Thank you.
Okay, thanks.
Great, thanks. All right. I think this is going to be a CDC question as well. Does the research show any variability in resistance in larvae versus adults of a species or of a given pesticide, meaning the resistance genes are expressed after metamorphosis?
Yes. So it depends on the metabolic resistance mechanism that is selected. For example, if it's high levels of oxidase, those are expressed evenly, relatively evenly throughout the entire life of the mosquito. If esterases are the basis of the resistance, esterases are expressed at their highest levels in the pupal stage. After that, adult emerges, the esterase levels drop off. And usually by about the time that mosquito is 14 days old, they look the same as susceptible mosquitoes. So that's another reason it's important to understand your resistance mechanism at play, because that has big implications in disease control. If you can't kill the young ones that aren't infected yet, that's not such a bad thing as it would be if you can't kill the old infected ones. So it's very important. And Mark touched on blood feeding as, you know, when a mosquito blood feeds, when it takes a sugar meal, all of these enzymes are turned on to digest that blood and to make eggs. And so when you have overexpression of enzymes, they can also affect exposure to insecticides. So that's why it's very important when you do your assays, to the best of your ability, to know the age of the mosquito that you're testing and the physiological status of it.
Great, thank you for that. And since you mentioned Mark, we'll go ahead and put him in the hot seat. We have a question here for him. Are blood-fed gravid mosquitoes less exposed to adulticide due to limited flights?
Yeah, I mean, that's in our natural kind of field population. That's one interpretation of the lack of control. But I think some of those laboratory studies where there's been topical applications have shown that there is an actual physiological or molecular mechanism at work as well. So both could definitely be at work, which actually just exacerbates the whole situation.
Right. Yeah. All right. Well, we do have a few more questions, but we're kind of limited on time. So we're going to go ahead and wrap it up because we want to make sure that we get all your CEU information and the secret code word that everyone is dying to know about. We're going to provide that for you here now in our closing. So thank you all for taking the questions. The leftover questions we did not get to, we'll put those together and we'll reach out to these presenters and get answers for each of you that had the questions we didn't get to. But thank you all for the questions and the presentations as well.